to deliver her inaugural speech. And her words have been always a source of inspiration for most of us. So over to Professor Sarkar.
Respected speaker, Dr. Budhaditya Mukherjee, our organizers, that is the Postgraduate Department of Zoology, Lady Brabourne College, headed by Dr. Sangjukta Mandal Parui, our viewers, that is the participants who are participating in today's webinar. A very good morning and a warm welcome to all of you. It's a wonderful occasion when the Postgraduate Department of Zoology of Lady Brabourne College is organizing a one-day webinar on a very interesting topic, understanding the mysteries of the zoological world for creating a better future. Here, Professor Mrinal Kanti Ghosh will speak on human health and cancer, and Dr. Mukherjee will speak on survival strategies opted by intracellular protozoa pathogens to evade post-immune response. Under the present circumstances in the pandemic situation, these two topics are quite timely. These are all hardcore biological science related topics, and science is never my cup of tea. So I just want to convey to you that we as human beings, we are all dream-loving people. We all want to dream of a better future, a more sustainable tomorrow. I can remember Tagore's very famous poem, Shobhotar Prati, that is to civilization, where he utters these words, Dao fire she aronyo, lao e nagor, lao jato louho lostro kashto prostor. That is, get me back that old forest, take back the urban civilization, take back all iron, wood and stone. And he concludes the poem with these words, Chai Shadhinata, Chai Pokkher Bistar, Bokkhe Fire Pete Chai, Shokti Aponar. Koraner Sporshite Chai, Chhiriya Bandhon, Anantoe Jagoter Hriday Spandon. That is, we want freedom. We want the expansion of our wings. We want to get back our old strength in our hearts. Tearing all strings of bondage, we want to touch the pulse of this world. So this world is, to Tagore, is a living thing. And we also want to make it a living one. But you know that a poet is only a dreamer. But a scientist is something more than that. He is both a dreamer and a designer, an implementer. He can translate his dream into a project, into a plan, a concrete project and a plan. So we will expect from the scientists that there should be a better future, a more sustainable tomorrow. We really do not want to get back to the old days of forest. As individuals, we all want urban facilities, just like the rural people also opt for urban facilities. But we want a more wise fusion between urbanization and eco-friendliness. We want a more balanced blending between all kinds of urban facilities and environment friendliness. I'm not going to, take more time by lingering my speech because I'm not an expert in this field. We are very eager to know from the experts. But I'm pretty certain that after both talks, there will be lots and lots of questions raised by our viewers today. And lots of their probable answers too are to be provided by our speakers today, Professor Ghosh and Professor Mukherjee. And at the end of the webinar, I am pretty certain that everybody will be able to know a lot more new things and a lot more new ways of explaining them. Everybody present here will feel more and more enriched and more enlightened. I wish today's one-day webinar a grand success. Thank you.
Thank you, Professor Sarkar. Wonderfully spoken. And now may I request Dr. Sanjukta Mondal, Head of the Department of Zoology, Lady Brabourne College, to say a few words. Thank you. Over to Dr. Sanjukta Mondal.
Thank you, Poulumi. Very good morning to all of you. Honorable Principal, Professor Shuli Sarkar. Our honorable speakers today for today's webinar, Professor Mrinal Kanti Ghosh from IICB Kolkata and Dr. Budhaditya Mukherjee from IIT Kharagpur, all teachers, students, and all our distinguished participants today. It is a proud privilege for me to extend my heartiest welcome to you all to this webinar. Now, understanding the mysteries of the zoological world for creating a better future. This was our theme. This is our theme today. The advent of all modern techniques and tools have allowed us for isolating and characterizing biomolecules, which allows a molecular approach to essentially all biological problems. An inquiry into the individual animals and their constituent parts, even to the molecular level, and the inquiry into animal populations, entire fauna, and the relationships of animals to each other, to the plants, and to the non-living environment. Though this wide range of studies have resulted in some isolational specialities within zoology, the conceptual integration in the contemporary study of living things that has occurred in recent years emphasizes the structural and functional unity of life rather than its diversity. The new innovations in the field of biological science are really remarkable and worthy for mankind, both for treatment and medicines, for the various deadly diseases for which the medicines, the treatment has been formulated. So the theme for this webinar is a topic that is of considerable interest, not only for our students, but as well as for the research community. And frankly speaking, this is quite evident from the overwhelming response I got from the participants. We had planned this webinar on a rather small scale to accommodate around 250 of the participants, but the response was so huge that it motivated and inspired us to organize this. We are planning of organizing similar seminars in future on a larger platform so that we can accommodate more number of participants. For this webinar, we have accommodated the extra participants through live streaming, and I'm sure all will feel enriched with the knowledge after completion of this event. Our recorded version of this lectures will be available on YouTube or Facebook, which we will intimate later, so that you can refer to these lectures later on. And after the process of registration was over, I found that nearly 70% of the participants are students over here. So I requested my distinguished speakers to speak more on, a little bit more on the topic in such a way that the students are more benefited, so that they can correlate this topic with their syllabus, they are benefited, not going much into the research angle. So, and they have agreed to this, and they said that, yes, of course, they will just keeping this in mind, they have formulated their lectures. So on behalf of the department, postgraduate department of zoology, Lady Brabourne College, I again welcome you all to this webinar, and I'm sure you all will be having a great time listening to our speakers. Thank you so much. It's over to Polomi.
Thank you, Dr. Mondal. It's time for the first deliberation, ladies and gentlemen. And it's a privilege to have Dr. Budhaditya Mukherjee, Assistant Professor, IIT Kharagpur. May I now request our undergraduate student, Ms. Ananya Mondal, to kindly introduce Dr. Mukherjee. But before that, let me request the participants that please keep your questions for Dr. Mukherjee and for Dr. Ghosh in our chat box, so that we can collaborate them in the question answer session. Thank you. Over to Ananya.
Good morning, everyone. I am Ananya Mondal, and it's an honor for me to introduce Dr. Budhaditya Mukherjee. Dr. Budhaditya Mukherjee is presently serving as an assistant professor at the School of Medical Science and Technology, Indian Institute of Technology, Kharagpur. He did his graduation and then master's degree in genetics from University of Calcutta. He then did his PhD from the Department of Infectious Disease and Immunology, Indian Institute of Chemical Biology, before joining as a postdoctoral fellow at the Department of Microbiology and Immunology, Johns Hopkins Bloomberg School of Public Health in 2014. He later shifted to the Department of Microbiology and Molecular Medicine, University of Geneva, as a postdoctoral fellow in 2015, where he continued till 2019 before joining IIT Kharagpur in the same year. He has several high-impact peer-reviewed publications in various national and internationally reputed journals. He also has several awards and fellowships to his credit. These include Parafrap International Fellowship in 2014, EMBO Long-Term European Fellowship 2014-15, Swiss Government Excellence Scholarship 2015-16, INSA Medal for Young Scientist in 2018, Pfizer Research Award in the area of infectious diseases, rheumatology and immunology in 2018. He is presently working in collaboration with UCB Celltech UK, a branch of UCB Pharma, SA and Medicines for Malaria. It is over to you, Dr. Budhaditya Mukherjee.
Okay. I have been already been introduced by Ananya, and it's a nice introduction. Thank you. But as Professor Padu already mentioned that this has been requested for a student, and I expected that this will, I have modified my lectures such that it is understandable to and appealing to them. However, if you have any question, please ask me. If you don't understand anything related to the presentation or the techniques that I talked about, please let me know, and I'll be happy to answer. So today's topic is survival strategies opted by intracellular protozoan pathogen to evade host immune response. So let me just give you a brief background. I have already been introduced, but I thought that if I talked about this, you will be able to understand the career path that I followed. So I did my master's in genetics in 2008, and this is from CU. And after that, I joined CSIR ISCB as a PhD candidate in infectious disease and immunology division. And at this point of time, I was working on Leishmania donovani parasites. mostly on their host-parasite interaction, particularly in context of drug resistance. So I completed my PhD degree and submitted my thesis around 2014. And then unfortunately, I have to leave Kolkata and I have to go to postdoctoral studies in US, and I joined John Hopkins. I was in that phase working on the colitis model, and I was working on the innate immunity in the colitis model. But however, I wanted to learn a bit more, something more than my immunology career in the parasitic disease or infectious disease, although my broad goal was to keep myself aligned in infectious disease. With this view, I joined Dr. Dominic Soldati's group in University of Geneva and started working on epicomplex and parasite like Plasmodium and Toxoplasma, more on the cell biology and biochemistry aspect of these parasites. And then finally, I joined 2019. In 2019, I joined IIT Kharagpur in the School of Medical Science and Technology. And I have been now planning to work and started my lab focusing on both of this kinetoplastid and epicomplex and parasite leishmania on the different aspects of immunology, but not only immunology. Like I wanted to combine my postdoctoral and PhD knowledge. So I'm working on both on cellular and biochemical aspects of these parasites. So if you look in the direction of my research path, okay. So as I was talking that I will not talk about this drug part, which has a overlap of my PhD and postdoctoral studies. And then in my postdoc, I mostly work on biochemical and cellular aspects of epicomplex and parasite. Now, you can see my research path and appreciate the fact that I started from host immune response and then went to parasite. But unfortunately, the parasite doesn't follow the same path of infection. To infect a host cell, it first must come in contact with the host, establish its infection, and then encounter host immune response. So I have decided to arrange my presentation in such that I will be talking about this process of establishment of infection in the host first, and then I will go to host immune response with respect to the different protozoan pathogen. So for this cellular and biochemical aspects that allow this epicomplex and parasite, my model is mostly Toxoplasma and Plasmodium parasite. And for referring to host immune response, I'll be talking about Leishmania as a model. So let's look at this protozoan pathogens. We can see that there is Toxoplasma, Trypanosoma, Leishmania, and you can all appreciate the fact that these parasites first come in contact with the host cell, whether they can actively invade their host or taken up by receptor-mediated endocytosis. And that is the time point when they started to modulate the host. The main function of modulating the host is to establish a successful infection that allow this parasite to. So as I was talking about, you can see that this epicomplex and parasite not only infect human, but they also infect a large number of livestock, and that is also economically important to economically important to us. However, this is also important to mention that this is the largest group of protozoan pathogen which causes the maximum number of death in human each year. So I have chosen 3 of them: Plasmodium, which causes malaria and 500,000 deaths each year; Toxoplasma, a parasite which can infect about one-third of the human population, and Cryptosporidium, which in recent years has come up as one of the major cause of child death in India after the rotavirus. So let's see the infective stages of this parasite. Now, if you look at the infective stages of this epicomplex and parasite, be it a Plasmodium merozoite or a Toxoplasma tachyzoite or a Cryptosporidium sporozoite, you will see that certain specialized secretory organelle like micronym and rhoptry are present in all of them. Now, as in case of this parasite, as in case of any other epicomplexan parasite, the process of infection is a tightly regulated process which requires synchronized secretion from the specialized organelle like microneme and rhoptry, which contains several proteins like perforins, adhesins that allow these parasites to successfully invade their host and establish the infection. So what happens is these microneme proteins are released first. This allows this parasite to get out or lyse the host membrane from their existing infected host. And once they're out, they started the process of reinvasion. The first step of this process is attachment, and then the parasite orient themselves in a 90-degree manner with the host cell. And after that, they release their rhoptry content, which again can be divided into two parts, rhoptry bulk protein and rhoptry neck protein. So rhoptry neck proteins are released first. This helps in the formation of a tight junction or moving junction between the host and the parasite. And the rhoptry bulk protein are then released into the host, and this goes into the host nucleus and modify the host environment, thereby creating a perfect niche where this parasite can live and survive. So if you look at the process of the invasion in a video, you will look something like this, where the parasites started to glide. At some point, they get in contact with the host and started to enter the host slowly. Now you can see this, the formation of a moving junction as the parasite enters the host, and ultimately it completes the process of infection. Now, if you tagged a rhoptry neck protein during this process, you will see that this rhoptry neck protein during the process of invasion move into the moving junction, which again emphasizes the role of this protein.
Sorry, again you have muted yourself. Means somebody else is muting you. That's a problem over here. So, sorry, Dr. Mukherjee. Can you please continue from this slide?
So I guess you are clear with the video.
Yes. Okay, thank you so much.
So as I was talking, that like any normal protein, this microneme and rhoptry protein is also going through the secretory pathway, by which I mean the endoplasmic reticulum Golgi, and finally reaching their final destination in the microneme and rhoptries. Now, if we look into this process, what happens is in the secretory pathway, this microneme and rhoptry protein get proteolytically processed. And this processing is required for the proper functioning of this protein when they reach their final destination in microneme or rhoptry. We were able to localize an aspartyl protease that is specifically present in this secretory pathway, as you can see this in red, where this rhoptry and the microneme proteins get processed. And we decided to study the effect of this aspartyl protease on the processing of the microneme and the rhoptry protein, so as to have an impact on the overall lytic cycle of these parasites. Now, what are aspartyl proteases? So aspartyl proteases are the enzyme that are present in all eukaryotes. So normally they have a pro region and an active site which consists of a DTG or a DSG motif, and within the active site there is a flap and flexible loop which allows ligand binding. So they have a broad range of function which includes protein degradation, enzyme maturation, signal transduction, and they have been a preferred choice for chemotherapy, particularly after the success story of HIV-1 aspartic protease inhibitor. Now, if you look at the broad repertoire of aspartyl proteases in epicomplexan parasite, you can see that they can be clustered into several groups, each group assigned with a specific function. For example, most of you are probably aware of hemoglobin degrading plasmapsins, which are present in the food vacuole of the parasites. The aspartate protease that we localized in the secretory pathway is TG-ASP3 or Toxoplasma aspartate protease 3. And the function of that protein at that point is unknown. And it has two orthologs in Plasmodium, plasmapsin 9 and plasmapsin 10. So we decided to determine the function of this aspartate protease. So like any normal approach to determine the function of a particular protein, what you do is you get rid of the protein or you create a knockout of the protein. However, a repeated attempt from us to generate the knockout of this protein is not successful, suggesting that this protein are essential for the parasite survival. How we detect this? We detect this with the help of a simple assay called plaque assay. Now, as you can see, as I have already mentioned, the parasite invade the host and then it stays and replicate within the host for some time. And after that, at some point, it decided that it had enough and now it wanted to get out of the host and invade a new host. By doing so, it lyses the host membrane, and if you stain with crystal violet, you can see this lysed form of the host cell as plaques that will appear in this white form. Now, if you have a defect in any point at this lytic cycle of this parasite, be it intracellular growth or egress or invasion, you will see that these plaques are not forming. So, in case of the wild-type parasite, you see that this clear formation of the plaques are taking place. However, if you knock out the protease, you see that there is no formation of the plaques, suggesting that these proteases are essential for the survival of the parasite. This prompted us to opt for an inducible knockdown system, meaning you can switch on and switch off the expression of the protein to study its function. So for that, what we did is you have an endogenous promoter for this particular protein ASP3, and for this particular gene ASP3, and you replace this endogenous promoter with a TATE promoter. We use a CRISPR-Cas9 and guide RNA to replace this endogenous promoter with a TATE inducible promoter. So under normal circumstances, this promoter is active and the mRNA is produced and it eventually gets translated into protein. However, if you add anhydrous tetracycline, the promoter is switched off and there will be no more formation of the ASP3. As you can see here, in minus ATC condition, which basically represented the wild-type condition, you can see the protein formation like its pro form and finally its processed mature form. However, if you treat with ATC, then you see that this protein is not forming anymore. As you can see, the disappearance of the protein, both in a Western blot and also from the immunofluorescence, you see that this red represents the ASP3. And if you treat with ATC, this protein is gone. So the first thing that we did is to study the effect of this knockdown on the processing of the ROP3 and micronym protein. Now, you can see that in case of the wild-type parasite, MYC6, which is a representative of a micronym protein, get processed from its pro to mature form. Okay, so around 55 to 40 kilodalton. Now, if you treat with ATC to create the knockdown, you see that there is an increasing accumulation of MYC6 unprocessed form. Same is the true for the ROP3 protein, where in case of the wild-type parasite, you see the mature form of the protein appearing. However, if you knock down the protease, you see increasing accumulation of the unprocessed ROP3-1 protein. And MYC6 and ROP1 used here are just two representative of the micronym and ROP3 protein. This is true for many others micronym and ROP3 protein that we checked. So you can see from this, there is an accumulation of unprocessed micronym and ROP3 protein in case of the knockdown parasite. So the first thing that we investigate in this direction, what is the effect of this accumulation of unprocessed ROP3 protein on the lytic cycle of the parasite? Now this we did with the help of a simple assay, which we regard as evacuole assay. Now, as I have mentioned that during the process of the invasion, this parasite discharged their ROP3 content into the host that allow them to infect the host and also to modulate the host function. Now, if you treat with cytochalasin D, which is an actin myosin motor inhibitor, these parasites can still attach themselves on the surface of the host. and they can discharge their rupture content. However, they fail to move inside the host. And if you stain it with a rupture resident marker like ROP1, you can see something like this, where you see that in Y-type parasite, you have a rupture discharge. As you can see here with ROP1, the parasites are shooting their rupture. However, if case of the knockdown parasite, you see that there is no more rupture discharge, and most of the rupture content, as represented here in green, is within the parasite. So what this suggests that the accumulation of this unprocessed rupture protein result in a defective rupture discharge for this parasite, which eventually affect the process of invasion. So next is, what is the effect of the accumulation of the unprocessed microneem protein on the lytic cycle of this parasite? To do this, I will show you again a simple video, which is the best way to explain to you visually. Now, this is for the Plasmodium parasite. As you can see, for the Plasmodium parasite, the parasite, when they are in Y-type condition, they can rapidly rise their host RBC membrane and come out. As you can see here, they're lysing their host RBC and increasing number of the parasites are coming out. Now, for the visual representation, what happens in the knockdown, I have taken Toxoplasma parasite, which form a large parasitophorous vacuole. And as you can see here, these parasites are trapped in a large vacuole. They're trying to break this vacuole and come out of the host. Eventually, one of them do manage to do so, but most of them remain trapped in this ball containing vacuole. This is more clear here. As you can see here, the parasite is blocked in a ball-like structure and it fails to break the ball and go out. So there is a defect in egress. As you can see here, the accumulation of unprocessed microneem protein results in a defective egress or exit of the parasite from the infected host. So the two important components, invasion and egress, is defected as a result of knocking down of this aspartate protease. So if I summarize, you see that in case of Toxoplasma parasite, ASP3 helps in the process of invasion. Similarly, it also helps this parasite to get out of their host, a process called egress, and it acts as a maturase for the microneem and rupture protein, resulting in their processing from pro to mature form, which function in this process. This role of ASP3 is completed by two of its ortholog in plasmepsin, in Plasmodium, where plasmepsin 9 act as a maturase for the rupture protein and help in the process of the invasion, and plasmepsin maturase for both rupture and microneem protein and help in the process of egress. So moving on, now we will talk about host immunity. I guess that you have an idea how a parasite can infect the host and how it goes out of the host to spread the infection. Now let us look from the host point of view. Now we are looking from the host direction. So this is a generalized representation. So what happens is, any parasites, intracellular pathogen I'm specifically talking about, so when it comes in contact with the antigen presenting cell, be it a dendritic cell or macrophages, there are various pattern recognition receptors like TLRs, which can also be cytoplasmic, which recognize this parasite by the virtue of its various PAMs, which are maybe protein or lipid that are present on its surface. So what happens is, this triggers a signaling pathway in this host, which is by the process by which the parasite proteins are processed, either in the phagosome, where the lysosome fused with the parasitophorous vacuole, and once this parasite antigen are processed, they are represented as MHC2, which gives rise to a CD4 T cell response. On the contrary, in case of the virus, if you see that it can be represented also in MHC1, not exclusively only for virus, but also for pathogen, and it activates the CD8 T cell response. So this can be divided broadly into innate immune response and adaptive immune response, as I was referring to. So mostly for this presentation, I'm going to talk about how parasite can modulate the innate immunity of the host, as this is the process of initiation of the host immune response. Okay. Now, for this, I will be using Leishmania pathogen, Leishmania parasite as a model. I know you were aware of this. So Leishmania is a pathogen which can exist in a mastigote or a amastigote form. So this is a sandfly where the parasite exists in its pro-mastigote form, and this pro-mastigotes were injected into the healthy human being when the sandfly takes its blood meal. And once it's entered the host, it gets converted into amastigotes, and it goes into liver and spleen where the parasite mainly replicate. But at the same time, like all intracellular pathogen, they invade the host, and at the same time they come out of the host, which are then again picked up by the sandfly, and the infection circle is repeated, and this allows the transmission of this parasites. Now, for all the intracellular pathogen, as you can see, they are a master manipulator that allow them to modify the host in which they resides, just to avoid the host immune response. So a parasite's main goal will always be not to kill its host, but to devise a way by which it can survive within the host. For example, if we consider trypanosoma, you see that these parasites, when they enter the macrophage, they recruit lysosome on the surface of the membrane. This allows the formation of the pore by which the parasite can enter the host, and they stay within the intracellular vacuole, which is parasitophorous vacuole, for a short period of time, after which they goes out and exist in the cytoplasm. Now, another example of this will be Toxoplasma. You can see that these parasites perform an active entry that I have already mentioned and explained to you with the help of Lobtry and micronein protein or Plasmodium. So these parasites form the parasitophorous vacuole membrane actively, but they exclude the host component from their parasitophorous vacuole membrane. This allows this parasite to avoid them from fusion with the lysosome so that the phagosome does not mature. So what happens is that when the phagosome does not mature, there is no degradation of this parasite protein, and hence there is no MHC representation. Now I will talk a little bit more about Leishmania. Please ignore the complicacy of the figure, but what I wanted to mainly show to you that in case of Leishmania, it is taken up by phagocytosis. It's not an active entry, but it is a receptor-mediated phagocytosis. Now, once it's entered the host, the parasite have got several proteins like GP63 and LPG on its surface that allows this parasite's early endosome to modify themselves in such a way that they never get fused with the phagosome or the lysosome to form the late phagosome. So what is the effect of this? That this already prevent the parasite antigen from being processed. Now imagine if there is no antigen processing, of course there will be no antigen presentation, and in this way the parasite could evolve to escape the host immune response. Also, there is a problem by which this parasite can modulate the loading of the processed peptide, whatever it may be processed, into the MHC complex. Meaning there may be functional peptide which get processed, but it is never loaded into the MHC. They can also reduce the expression of the MHC. And finally, they alter the membrane of the infected macrophages or infected host by quenching cholesterol from them. Thus, whenever it's represented on the surface of the antigen presenting cells, be it a macrophage or a dendritic cells, the signaling, the proper TCS signaling does not, T cell mediated signaling does not take place, as this receptor, as this MHC representation of the peptide is not stable due to the fluidity of the membrane. So these are the primary ways by which a Leishmania parasite can evade its host immune response. Now for India, the problem is a little bit more complicated, because when we talk about Leishmania in India, we are not talking about only the parasite, but we have to consider the drug resistance factor. Now, if you see, this is a picture of hyperendemic zone of Muzaffarpur, hyperendemic zone of UP mostly and Bihar, and you can see that most of these parasites coming from this region are resistant to the antimonial drug. So antimony was used for the treatment of leishmaniasis long back, around 1921 by UN Brahmachari, and with its introduction, the drug was a dramatic success, but with the time, the drug gradually loses its efficacy, mostly due to the emergence of the resistance. And with this, new drugs like antimonials, the new drugs like meltaphosin, amphotericin B came into picture, but the resistance against this drug also emerged at a rapid rate. So a relevant problem for this Indian subcontinent, particularly and also from the other part of the world, is the emergence of the drug resistance. And if you look at this region, the clinical isolates that are obtained from this region, around 77% of them are resistant to antimonial drugs. This can clearly give you a picture of the spread of the antimony resistance. However, importantly, if you look at the biochemical and biophysical properties of these parasites, you will see that apart from their different responsiveness to the antimonial drugs, this parasite also differ in large number of biochemical and biophysical parameters. For example, they express different sugar on their surface, they have different membrane fluidity, they induce the host transcription factors differently. So basically, this is not very hard to imagine that these parasites, which differ so much in their biochemical and biophysical property, can modulate their host cell differentially, thus ultimately resulting in an entire different disease outcome. So the major question that we try to answer in this direction is why the infection with the antimony resistant parasite always results in an aggressive pathology. Now, there are several reports starting from 2003 from Bihar from Dr. C.P. Thakur that mainly highlighted the fact that the patients who were infected with antimony resistant parasite always results in a high parasite load. So how antimony resistant Leishmania parasite can modulate their host, resulting in an aggressive pathology and a different disease outcome, was the main question of our study. And all the antimony resistant parasite mentioned in this study are obtained from the hyperendemic zone of Bihar. Now, as I have mentioned to you, that one of the major factor that they modulate the host cell differently, and this has been reported long back. Like the first report was in 2003 when Dr. C.P. Thakur observed that the patient harboring antimony resistant parasite always results in high organ parasite load in their liver, meaning they have a high load of the parasite, and which is always correlated with high level of IL-10, interleukin 10, which is an anti-inflammatory cytokine in their serum. Now, for these clinical isolates, we observe the same observation, which has been also reported by others. We saw that all these antimony resistant parasite, as you compare with the blue, which are antimony sensitive, and red, which are antimony resistant parasite, which infect the host, you see there is an increased production of IL-10. And this is a consistent fact. And we really decided to study what is this role of IL-10 in this organ parasite burden. So how the antimony resistant parasite can induce this high level of IL-10 in their host. To study this, if you look at the PubMed and search that how IL-10 level or IL-10 can be modulated or host IL-10 can be modulated, you will see that the number of papers come out which suggest different pathways for this particular gene to be regulated transcriptionally or post-transcriptionally. So what we did, we choose inhibitors from this different pathway and started to apply them one by one or in combination, and observe the effect of the production of the IL-10 with respect to antimony resistant and antimony sensitive infection. As you can see from our study, as compared to the untreated control, 3 inhibitors, which are mainly ERK, PI3K inhibitor and NF-kappa B inhibitor, we see that there is a drop in IL-10 level or antimony resistant parasite induced IL-10 in the host. And this is also corroborated with the fact with the signaling pathway that this parasite generate. So when you infect the host cell or the host macrophage with this resistant parasite, you see that these inhibitors of ERK, ATK and NF-kappa B, you can detect actually the phosphorylated form of this ERK, NF-kappa B into the nucleus of the host. As you can see here, the ERK phosphorylated form is going into the nucleus of the host. It is also corroborated with the degraded phosphorylation of IKK alpha, IKK beta, basically suggesting that this ERK NF-kappa B pathway is the critical pathway which is induced by this antimony resistant parasite to regulate the IL-10 level in the host. So NF-kappa B is a transcription factor. It's widely used, control the transcription of many human genes. So when we look into the IL-10 promoter, we find that the IL-10 promoter have got 3 NF-kappa B sites in it. So these are the 3 NF-kappa B sites in the IL-10 murine IL-10 promoter. And we started to prove our concept that NF-kappa B is the transcription factor which is binding to this promoter site to regulate the IL-10 level in the antimony resistant infected host. We started to delete them one by one. So basically we generated the deletion construct and also perform a site-directed mutagenesis. We find that the second site, which is 583 to 593 in the IL-10 promoter, is the principal site which regulates the IL-10 level in response to antimony-resistant parasite infection. As you can see here, if you have the entire IL-10 promoter, you have a strong induction of IL-10 in response to antimony-resistant parasite, which in this case is represented as BHU-138 and BHU-575. However, if you delete this promoter, there is a drop in the IL-10 level. Similarly, if you just mutate this promoter rather than deleting it, keeping the other 2 promoter site intact, you still see this significant drop in IL-10, which clearly suggests that this IL-10 promoter site is the critical for the NF-kappa B binding. We further prove this with the help of a chip and the EMSA assay. So what we do for the chip assay is we observe whether a transcription factor is getting epigenetic, whether the promoter site of a gene is getting epigenetically modulated that allow the transcription factor to bind with it. Now, in case of antimony resistant infection, if you compare with the sensitive or the uninfected cells, you will see that you can detect acetylation of the histone of the IL-10 promoter and also the recruitment of the NF-kappa B subunit P50 and CREL into the IL-10 promoter with respect to antimony resistant infection. And you can also detect in the nuclear fraction that P50 phosphorylated form is present. CREL, which is another form of NF-kappa B, basically subunit, which form the heterodimer, is also present. So this clearly suggests that in response to antimony resistant infection, this NF-kappa B subunit P50 and CREL get translocated into the host nucleus and it binds on a specific site of the IL-10 promoter. Now, if you take the site 2 of the IL-10 promoter, which is have a sequence of this, -583 to -593, which we say this as a Y-type murine IL-10 probe. And you can see that with this murine IL-10 probe, if you radio level this murine IL-10 probe, and if you provide the infection or the parasite lysate, you can see there is a strong binding of this wild-type probe with the in response to SBRLD infection, which is absent if you take the mutant IL-10 probe. So basically, this clearly confirms that this sequence is important for the binding of the NF-kappa B subunit, which is further proved by a supershift assay. So for the supershift assay, what we do is we take the radioactive probe, as you can see here. So when there is nothing, there is a free run of the probe. Now, if you add P50 antibody, you will see that this P50 antibody is resulting in a shift of the probe. Okay. Now, if you add a P-CREL antibody, you see the same shift. And if you add both the antibody, you will see a super shift of the probe, which clearly suggests that as this antibody is binding with this Y-type probe, they are resulting in the formation of a higher molecular weight and resulting in the retention of the probe at a higher size in the EMSA gel. Clearly, you can also see the translocation of this P50 and CREL into the host nucleus. So P50 here is stained with green, CREL is stained with red, and the W basically represent the nucleus. And you can see here that these subunits clearly move into the host nucleus or translocate into the host nucleus with response to antimony-resistant parasite infection. Now, I hope that I have convinced you that this signaling pathway, which is uniquely activated in case of antimony-resistant parasite infection, but not in case of their sensitive counterpart. Now, interestingly, in context with the antimony-resistant infection, if we compare the antimony-sensitive parasite infection, you will see that this several host machinery pathway, including ERK NF-kappa B, which we detected for the antimony-resistant parasite infection, are deactivated, meaning that this transcription factor are not active in case of antimony-sensitive parasite infection, which again suggests that these parasites have evolved a different way to establish their infection in the host, and they modulate their host differentially, thus resulting in an entire different disease outcome. Now, as I was mentioning to before about the different biophysical and biochemical properties, one of them is that this antimony-resistant parasite express a unique sugar on their surface or a unique glycoconjugate on their surface. The next thing what we wanted to relate is whether this expression of this glycoconjugate have any role in induction of the IL-10 in the host, or whether this glycoconjugate can specifically activate the IL-10 induction pathway in case of antimony-resistant infection. So you have already seen that in case of the compared to the antimony-sensitive parasite, if you infect them with the antimony-resistant parasite, you have a high level of IL-10 production. And you know that this antimony-resistant parasite express a unique glycoconjugate on their surface, which is not present in the antimony-sensitive parasite. So what we decided to do is we get rid of this glycoconjugate. And for this, we knock down a gene called galactosyl transferase 3 or GALT3, which is mainly responsible for addition of this glycoconjugate into the surface of the parasite. So what happens if we knock down this glycoconjugates? If we knock down this glycoconjugate, you can see there is a clear decrease in the IL-10 level as compared to the wild-type parasite. The knockdown parasite produce much more less IL-10 in response to infection. And this is more or less corroborating with the sensitive, antimony-sensitive parasite infection. And this is also reflected in the parasite load or the organ parasite load. A decrease in the IL-10 results in a decreased organ parasite load in case of the knockdown parasite infection. So we see that a sugar or a glycoconjugate on the surface of the parasite might regulate the IL-10 level in the host or regulate the host transcription pathway in such a manner that it helps or favors the parasite load. Now, since a glycoconjugate comes in, it is clearly observed, it was obvious to us a TLR or toll-like receptor, which are normally responsible for recognizing this glycoconjugate on the proteins on the parasite surface, might regulate the signaling process. So normally what are the TLRs supposed to do? So when a TLR recognizes a pathogen or a TAMP present on the pathogen, it initiates a signaling process which is mostly leading to a Th1 inflammatory response. Let's say IFN gamma response, which is an inflammatory cytokine. This cytokine basically kills the parasite and results in its clearance. However, parasites have evolved the way so that they can modulate this TLR signaling in such a way that rather than the killing of the parasite, it helps in survival of this parasite inside the host. Now, you can see that there are several TLRs. So it was not very obvious for us to which TLR to try first. But since this is a glycoconjugate, we thought that we'll start with TLR2 and TLR4. To do this, what we did is we shifted our experiment to knockout mice. So TLR2 knockout, we took TLR2 knockout mice and TLR4 knockout mice and infected them with antimony resistant or antimony sensitive parasite. Now you can see that in case of infecting the TLR2 knockout mice, as compared to the wild type C57BL6, the knockout mice were in this background. So this was used as a control. You can see that there is a clear increase in the amastigote load as compared to the resistant parasite in the wild type parasite. And this amastigote load is much less in case of the TLR2 knockout mice. However, TLR4 knockout mice have no effect in this respect. This is also reflected in terms of IL-10 production. Now, if you infect this TLR2 knockout mice with antimony resistant parasite, you see that in case of the TLR2 knockout mice, there is a drop in the IL-10 production, which is not in the case of TLR4 knockout mice, although you see some defect, but it is not as pronounced as TLR2. And this is also reflected if you take the infected liver and spleen from these infected mice. You see that the hepatic liver load, hepatic parasite load for this TLR2 knockout mice, as you can see here, is compared to the wild type mice is much less, and it is both a hepatic liver load is decreased, splenic liver load is decreased, and this also reflected in reduced IL-10 level. Furthermore, if you do a co-IP or co-immunoprecipitation assay. So there are several reports that the TLS can function in a homodimer or heterodimer, and we wanted to see what is the case in our case. So if you infect with BHU 575 or BHU 138, which are antimony resistant parasite, and if you do a co-immunoprecipitation, meaning if you probe down with TLR2 and then apply the TLS6 antibody, you can see that it can detect the TLR2 antibody in presence of, it can detect the TLS6 if you immunoprecipitate with the TLR2. So the basic purpose is to infect the cell and then apply the pull down. You do a pull down with TLR2 antibody and then you probe with TLS6 antibody to see if they are present together or not. And you can see clearly for the resistant parasite infection, you can see that there is a presence of TLR2, TLR6 heterodimer. And if you do the same with the TLR2 and TLR1, meaning if you pull down with TLR2 antibody and you try to probe for TLR1 antibody with respect to resistant parasite infection, you don't see any appearance of the TLR1 antibody. So from this, we conclude a model that antimony-resistant parasite express a unique sugar on their surface that allow them to specifically interact with host TLR, resulting in a heterodimerization of TLR2 and TLR6. And this results in the effective activation of the ERK NF-kappa B pathway, resulting in the translocation of P50 serial, which then bind to a specific site of the IL-10 promoter. Now, as a result, the IL-10 transcription increased, which results in a huge production of IL-10, and I have not shown this part. So this IL-10 can act in an autoregulatory manner, thereby increasing the expression or activation of another transcription factor, which in turn activate the overexpression of MDR1. Now, MDR1 is a MTPC transporter which is expressed on the surface of the cell, and this results in the efflux of the antimony from the host cell, thus providing a selective survival. Here, RH123 is a surrogate marker for antimony. So this results in the efflux of antimony and thus provide a selective survival advantage to this parasite to survive in presence of antimonial drugs. Now, what is the clinical importance of this study? So the clinical importance of this study is that you have seen that with the increase in IL-10, there is an increase in MDR1 transporter or APC transporter that can efflux the antimony outside the host. As you can clearly again see here that if you treat with antimony, if you infect with antimony resistant parasite, you can see that the macrophage are expressing MDR1 transporter on their surface. And if you treat this with the recombinant IL-10, you see that same. Without infection, the recombinant IL-10 can itself produce high expression of MDR1 transporter on their surface, high expression of MDR1 on their surface. And if you, so we are able to design small molecule inhibitors that specifically target this IL-10 mediated MDR1 activation in the host. And by that, we can make this antimony resistant parasite, which does not respond to the antimonial drug, which are now resistant to antimonial drug, to be susceptible to antimonial drugs once more. So this basically concludes how this study have clinical implication on the antimony resistance. And this, I think, will be all about drugs I'll be talking here. Now, there is an interesting story of this host-parasite interaction which I would like to share with you. Now, as I have mentioned to you previously, this antimony-resistant parasite, as they infect the host, they result in a heterodimerization of TLR2 and TLR6 in our case, and that basically activate the signaling pathway in the host that result in the production or over increase IL-10 level, and which in turn regulate the MDR1 expression and thus provide a selective survival advantage for this parasite. Now, if you see the TLR pathway, at that point we wanted to address the downstream molecules that are present for this TLR signaling. If you look at this, that this signaling can occur either in an immediate, which is an adapter molecule present inside the cell, in an immediate manner or immediate independent manner. Okay, so what you expect is if you knock down the immediate, you should stop the signaling, and then there should be no IL-10 production, and the antimony resistant parasites can be made susceptible to the antimonial drugs. So with this idea in mind, we decided to try our luck on immediate knockout mice, and we infected the immediate knockout mice with antimony resistant parasite, and we expected a reduced IL-10 level. But with what we observed was something very unexpected. We observed that if you knock down immediate, again C57BL6 represent the wild type mice, and immediate minus is the knockdown mice. If you observe that if you infect this immediate knockout mice as compared to the wild type C57BL6, you see that as compared to the wild type mice, there is a more IL-10 production and amastigote load or the parasite burden for these mice, as opposed to being downregulated in terms of IL-10 level or parasite lead. So at that point, we were very surprised and we didn't pursue this research further. But later on, when the initial paper got published, we decided to address this issue further and we started investigating in more details. Now, contrary to the IL-10, if you look at the TNF-alpha or IL-12 level, which are pro-inflammatory cytokine in nature, you will see that as compared to the C57BL6 and immediate, in case of the antimony-resistant parasite infection here highlighted in red, you will see that they also have a decreased IL-12 level, and which is even less than sensitive parasite infection. So we were confused as what is happening here, because we thought that immediate must be a critical molecule that regulate the IL-10 pathway, but it seems that it is not the case. Rather than suppressing the IL-10 induction, it's basically influencing the IL-10 production in case of antimony-resistant parasite infection. So to study this, we have a two hypothesis model. In the first hypothesis, we propose that the interaction of the antimony-resistant parasite resulting with the host TLR, resulting in the heteromerization of the TLR2 and TLR6, which in turn results in a decreased IL-12 level in the host, as you can see here. This is probably because of this. And then this decrease in IL-12 level is responsible for induction of IL-10 in the host, which in turn degrade the immediate. So immediate might not have any role in initial infection, but later on. The second model will be opposite, where the initial interaction of the parasite with the host receptor will induce the IL-10 production, and this IL-10 in turn can inhibit the immediate and IL-12 level in the host. So we wanted to distinguish what is actually happening. So to do this, we first check the immediate mRNA level in the infected host. Now, to our surprise, we saw a change in IL-10 level in immediate knockout mice, but if you infect it with the sensitive or the Y-type parasite, sorry, I forgot to mark them in red here. So these two, the resistant parasite, you see that with response to sensitive and resistant parasite infection, there is no change in the immediate, at least at the mRNA level. Even it remained mostly in the form of an uninduced level. However, if you infect with LPS, which is a positive control, you can see that there is a huge level of immediate mRNA expression. Now, if you repeat the same experiment and check the level of the protein, you will see for the resistant parasite, although there is no change in the mRNA level, you see that in case of the resistant parasite, there is no detection of immediate at the protein level. To further answer our question, if you treat the sensitive parasite with recombinant IL-10, you see that there is no alteration of the immediate protein expression, meaning the immediate protein expression remains same in presence of the recombinant IL-10. So we can clearly say that the excess IL-10 is not having any effect on immediate. Similarly, for the antimony resistant parasite infection, where you don't detect any immediate, if you started treating them or infecting them in presence of IL-10 neutralizing antibody to see if the IL-10 is the main reason which does not allow the immediate to express, you will see that increasing concentration of the IL-10 have no effect in reappearance of the immediate, and beta-actin is used here as a loading control. So you can clearly conclude that SBRLD infection regulates immediate expression post-transcriptionally, because the mRNA level there is no change, whereas in the protein level you don't see the appearance of any protein. However, it is also clear that the SBRLD induced IL-10 does not play any role in regulating the immediate expression. So having this clear in mind, we know that the immediate expression was regulated first and the IL-10 have probably got nothing to do with that. So what we did is, since we have an indication of the post-transcriptional regulation of the immediate, meaning it is not regulated at the mRNA level, but at the protein level, we perform a microarray for the microRNAs, which are basically short hairpin RNAs that can bind with the 3' UTR of a particular mRNA of a particular gene, and thereby block it from getting translated into the protein. So if you compare the microRNA expression profile in compared to SBRLD and SBS infection, antimony sensitive infection, you see a particular microRNA along with several others which is significantly expressed in SBRLD but is absent in LPS or antimony sensitive parasite infection. Many microRNA have this kind of profile, which are not represented here. But what interests us most in this microRNA is that if you look at the 3' UTR of the immediate, you see that this microRNA has got binding site in the 3' UTR of the immediate, suggesting that this microRNA can probably regulate the immediate expression by binding with its 3' UTR. So this is a microRNA that we understand that regulate the immediate expression in antimony-resistant infected host, as this is specifically upregulated in antimony-resistant parasite infection. So we started to looking into this microRNA and mechanism of its action into deeply. So if you look at when I started to look at this promoter of this microRNA with the promoter scan software, I couldn't find anything. Like, I was looking for 1,000 kb upstream of the promoter and I couldn't find anything. There was no promoter actually. Finally, I go on scanning and like 5 kb from the start point of the transcription, I find a promoter which perfectly matched the prediction score according to promoter scan. And I designated this so far as a hypothetical promoter for the microRNA 466I, which is basically present in 5 kb upstream of this gene. This kind of this kind of regulation do happen. So it was not a bit surprising. So when you look at this promoter, you see that this promoter is enriched with NF-kappa B transcription factor binding sites. So this was the most significant sites which qualify for the cutoff that this promoter have a significant NF-kappa B binding sites. Okay. So there were 3 NF-kappa B binding sites which qualify the cutoff more than 0.05 to be predicted. And so we thought that this NF-kappa B transcription factor, which we previously have been seen to be activated in case of the antimony resistant infection, is also regulating the expression of this microRNA. And this in fact comes out to be true if you just treat with the NF-kappa B inhibitor in presence of antimony resistant infection. You see that the pre-microRNA without inhibitor, which have a significant expression, pre-microRNA of 466I, which have a significant expression without the presence of the inhibitor. In presence of the inhibitor, there is a NF-kappa B inhibitor, there is a significant drop in its expression. So what are the sites that are important for this microRNA to regulate the MYD88 expression, or NF-kappa B binding sites, how it can regulate this microRNA expression? So we started with deleting, as before, initial sites and then continue. We find that each of these sites have somehow contributed to the regulating the expression of the microRNA 466I. As you can see here, that in case of when all the 3 sites are present and you infect with a resistant parasite, you see an optimal expression as compared to the sensitive parasite here in white, which doesn't induce this microRNA expression, which also correlate with the microarray data that we have. However, if you go on deleting, there is no absolute decrease. However, if you go on combination and delete all 3 promoter NF-kappa B binding site, you see there is this significant difference between the induction of MIR466I and its absence. Interestingly, so far, what we have detected that for this NF-kappa B signaling pathway, which activate in response to antimony resistant infection, we have always checked that this IL-10 induction occur late in infection, meaning for a substantial amount of IL-10 to be detected from the host, you need at least 12 hours post-infection. However, this microRNA, we can detect it as early as 4 hours or 6 hours post-infection. And when we check into the NF-kappa B transcription factor, P50 and serial at this early time point of infection, like 4 hours post-infection, which we haven't done before, we saw that we could detect these transcription factors, P50 serial specifically, in the host nucleus, even after 4 hours of infection with antimony resistant parasite. However, this NF-kappa B, in this early point of infection, if you check the microRNA promoter 466I at 4 hours post-infection, you will see that there is a significant recruitment of histone, acetylation of histone, along with binding of P50 and serial in these early hours of infection. However, if you check the IL-10 promoter at the same time point, you will see that in case of wild-type macrophages, at 4 hours post-infection, there is no recruitment of histone acetylation or P50 and serial to the IL-10 promoter. In fact, if you knock out this MYD88 and IL-12, meaning if you take macrophages from the MYD88 knockout mice or IL-12 knockout mice and then perform the same infection, now you can detect significant recruitment of histone acetylation and P50 serial in IL-10 promoter even at 4 hours of infection. So to further validate this, we check the IL-10 level comparing the C57BL6 mice, MYD88 knockout mice, and IL-12 knockout mice. Now you can see that in case of wild-type mice, which is C57BL6, which is represented by the black circle, filled circle here, as I was mentioning to you, that this always get a peak around 12 hours post-infection. So basically, to detect something significant in case of antimony-resistant infection, you need at least 12 hours post-infection. However, in case of the immediate knockout mice and IL-12 knockout mice, if you check the IL-10 level, you can see that there is, even after 2 hours, there is a significant upregulation in IL-10 in absence of immediate and IL-12, which is also corroborating with this cheap data, which clearly suggests that even at this early time point, 4 hours post-infection, there is a significant recruitment of the NF-kappa B subunits in the IL-10 promoter in case of immediate knockout or IL-12 knockout. Similarly, if you look at the IL-12 level, you will see that in case of the immediate knockout mice, if you infect with the resistant parasites, which are the filled circles or filled square here, you will see that the IL-12 level throughout the infection in case of the immediate knockout remains at a low level. Whereas if you take the wild type mice, there is an increased upregulation of IL-12 around 2 hours, which remains same, and then suddenly it drops. So basically, what it suggests is that if you infect the if you infected the host in absence of immediate or IL-12, then there is an increased surge of IL-10 even from the early point of infection, which is not present in case of the wild type infection, if you infected the wild type host. So what was the consequence of this? If you see the survival of these mice, you will see that wild type mice, if you infect with the resistant parasite, it have a survival curve which is not resulting in the death of the mice, because normally the mice can clear the infection. So there is an increased parasite load, but it is surviving. However, if you do the same with the immediate knockdown mice, you clearly see that there is a drop in the survival pattern of this mice. So suggesting that knocking down of immediate is not beneficial for the host. This clearly suggests, even with the histogram picture, as you can see here, with the histology, that in case of immediate knockdown mice, if you postmortem them after their death, you will see that they have the infected granuloma which still persists. This is around 42 days of sacrifice. And you will see that this granuloma are not properly formed. So suggesting that there is no proper clearance of the parasite. On the contrary, if you look at the bulb C or the wild type mice, you will see that there is distinct granuloma formation, and these black dots which represent the parasites are missing from here. So what does it conclude? It concludes that the antimony resistant parasite, if I sum up the whole story here, in antimony resistant parasite, they express a unique sugar or unique glycoconjugate on their surface that allow them to specifically interact with host TLR that results in the activation of a particular signaling pathway, including ERK and NF-kappa B. So this NF-kappa B moves into the host nucleus around 4 hours post-infection, but at that time, it can only access the promoter of the microRNA 466I. So it binds with the microRNA 466I promoter, resulting in its transcription and the formation of microRNA 466I, which then bind with the 3' UTR of the immediate, resulting in its deactivation. This deactivation of immediate result in a drop in IL-12 level in the host, and as the IL-12 level is dropped, the NF-kappa B subunits P50 serial now can access and bind with the IL-10 promoter, resulting in huge activation of IL-10 transcription. This IL-10 in turn upregulate host MDR1 and results in influx of antimony from the host. And as this work was going on, as I have already mentioned to you, that antimony-sensitive parasite, which does not express the sugar on this surface, results in deactivation of the host transcription factor, including ERK and NF-kappa B. Thus, they have devised a different strategy to establish or their silent entry into the host, which clearly suggests that these two parasites, even belonging to the same species, only in response to the drug response, only differing in the response of drug resistance and other biochemical parameters, can modulate the innate arm of the immune system differently to establish a successful infection. I think I will conclude my presentation here. I think I'm already 45 minutes. So thank you for your attention, and please let me know if you have questions.
Thank you, Dr. Mukherjee. It was a wonderful deliberation and very lucidly spoken. We have a number of questions for you in our chat box. So to corroborate these questions, I may ask our postgraduate student, Ms. Jhinuk Mitra, to take over and conduct this question answer session. So over to Ms. Jhinuk Mitra.
Thank you, ma'am. Good morning, Dr. Mukherjee. I would like to thank you first for this wonderful session which you had with us. It was very interesting, especially with the videos which you showed in the beginning of your presentation. It was very easy for us to understand that way. So before moving on to the questions, I would like to apologize to the participants that we are really short of time here. So we'll be taking up only a couple of questions. So, Doctor, the first question I have for you is there any available biomolecule in the serum which can estimate the expression of NF-kappa B? Over to you, sir.
Well, serum NF-kappa B, well, you can detect IL-10 level in the serum, but NF-kappa B is a transcription factor that normally goes into the cell. So if you wanted to detect something in the serum, it have to be IgG in response to infection, or IgM, IgA also in some cases. But detecting NF-kappa B in the serum is not going to be easy. I mean, probably you cannot do that.
Okay, thank you, sir. The next question we have for you is, what are the primary causes of increased viral shedding due to stress on reservoir hosts for several viruses? Could you please shed some light on this?
Okay. So if you look at the viral immune response, so normally a virus, let's say more relevant to now is the SARS-CoV-2 RNA virus. Everyone is interested to know about that. So why not? So this RNA virus are released in the cytoplasm, right? In case cytoplasm of the host, and over there they have been, they will be recognized by TLS7 or TLR8, which are present in the endosome. Basically, this endosome, from this endosome, they also have other pathogen receptor molecule like other PPRs, like MAD, RAB, MAD, RIG, and so on. And normally, what should happen is they should be able to signal the host to activate the IFN gamma response. Basically, IRF3 transcription factor should go into your nucleus, and then it should bind with IFN gamma promoter and start the signaling and start telling the cell that we have a viral infection, come on, clear it. But virus, the strategy is different. Whatever may be the reason, let's not go into that, but whatever may be the reason, this initial IFN gamma surge is not detected in case of the SARS-CoV-2. I mean, if it has been a prominent response for those hosts who can clear the infection successfully, this IFN gamma response is there and they doesn't have any symptom. Most of them are staying asymptomatic and the viral load gets clear. Now, if this initial clearance does not occur, and let's say the virus survives without any detection of IFN gamma response within your host, it will go on multiplying. And at some point, you will have a huge amount of viral load. So basically accumulation of viral RNA and viral protein. Now, the lung cell or alveolar type 2 cell at some point will, because of maybe this load or other environmental, or as you can say, other stress factor, will activate a huge amount of response. Now, that will be presented in MHC1 and will attract a huge amount of macrophages, and which will take up this and presented them in MHC1, resulting in a CD8 T cell response that at that time will produce huge amount of pro-inflammatory cytokine, mostly IL-6. Now, this cytokine will not distinguish between your host and virus and started killing everything randomly. And that is the major cause of this death and associated morbidity. So most of the patient who are dying from this viral infection is due to this cytokine storm. And the cue for this is initial deprivation of the IFN gamma response, which is followed by a higher surge in pro-inflammatory response in late hours of, late point of infection, mostly around 6 to 8 days. So that will be the main reason.
Thank you, sir. Our last question for you today is, can you briefly explain how the ROF3 protein disrupts the cell homeostasis in host?
Homeostasis. Okay, so the ROP3 is around, it's not a very, it's not a very, its amount of protein content is not very big, but it's still quite some. So there are several ROP3 proteins that have been identified for their function, mostly by reverse genetics approach, where you can knock out a particular ROP3 protein and see if it has function in the host cells in the infection of the pathogen or not. The first one was ROP1, which I've shown in the presentation, that was detected in 1992 and was initially suggested to be important for infection in the host. So the ROP3 mostly plays a role in infection. So either ROP3 NEC protein or ROP3 bulk protein. So ROP3 NEC protein mostly is responsible for the initial infection where they form where they're commonly called as RON protein, where they form the moving junction. Okay, once this, if you disrupt this, the infection will never take place. So this, along with the microneem protein MA1 and the ROP3 NEC complex, this is the first step that the parasite do to get them attach itself in the host. Now, these ROP3s are like injection. So you can push the protein into the host cell when it get attached. So first there is a strong attachment, then you position itself like say shooting of a revolver or shooting of a gun. You point it and then you discharge your ROP3 content within the cell. So this goes into the host nucleus, and then they can modulate the host immune regulatory gene for toxoplasma infection, GTPase, which basically bind with the parasitophorous vacuole of the parasite and results in its destruction, or IFN gamma, which results in the destruction of the parasite and two main immune response from the host. So what this ROP3 protein do is they have several kinases like ROP16, ROP13, and ROP18. So this goes into the host nucleus and binds with the transcription binds as a transcription factor into those genes. So basically this kinase phosphorylated the transcription factor that this can activate those transcription factor and bind with the promoter of this gene to regulate their transcription. I think R16 is another one, but this is not a ROP3 protein, but it's a dense granule protein. But ROP16 has been most well-studied kinase in this regard. So basically, these are the kinases which have been studied so far, or the ROP kinases, which can really modulate the host immune response.
Thank you so much, sir. Thank you for being with us today and for this wonderful deliberation. I would now request, thank you, sir. I would now request Ms. Shinjini Ghosh, UG third year student, to introduce our next speaker for the session. Over to you, Shinjini.
Good morning, everyone present here. I am Shinjini Ghosh, and I am extremely thankful to all my professors for giving me this opportunity to introduce esteemed guest, Professor Dr. Mrinal Kanti Ghosh. Dr. Mrinal Kanti Ghosh, Senior Principal Scientist and HOD, Cancer Biology and Inflammatory Disorder Division, CSIR, Indian Institute of Chemical Biology, Kolkata, and Professor at ACSIR, New Delhi, did his BSc in Chemistry and then MSc in Biochemistry from University of Calcutta. He also completed his PhD from University of Calcutta before joining Lerner Research Institute, Department of Molecular Biology, Cleveland Clinic Foundation, Ohio, USA, for his postdoctoral work during 1997 to 2003. Later on, he joined as staff at Department of Cancer Biology, Cleveland Clinic Foundation, USA, and worked there till 2005. Since 2007, He's actively engaged in cancer research in cancer biology and inflammatory disorder division at CSIR IICB. His research interests include signal transduction, gene regulation, target identification, PTM and cancer therapy, targeted delivery of nanomedicine. His many high-impact peer-reviewed publications in various internationally reputed journals like Molecular Cell, the EMBO Journal, Journal of Clinical and Biomedical Sciences, Journal of Virology, Oncogene, Oncotarget, Cell Death and Disease, Cellular Signaling, Cellular Oncology, Journal of Biological Chemistry, Journal of Experimental and Clinical Cancer Research, Bioassay, Signal Transduction, and Targeted Therapy, etc. At present, he is in the reviewer and editorial committee of many journals as well as international research proposals like World Cancer Research UK, Health Research Board Ireland, and DBT, DST Serve Manu Mission and CSIR at the national level. He became a Fellow of the National Academy of Sciences in 2015 and Fellow of West Bengal Academy of Science and Technology in 2015. He is also the Bharat Vikas Award 2019. A very big welcome to you, sir, and thank you for gracing us with your presence today. And now I would like to request Dr. Ghosh to take over for his lecture session. Over to you, sir.
So this is a good opportunity from a college that I can share my research outcome from the for the new generation students who will one day come to that in the research community. So today the title of my talk is Human Health and Cancer. So I am going to tell this towards the scenario that everybody think about that cancer is a dangerous disease. It's a disease or it's a disease of multiple cause. But the thing is that everybody are thinking that you can survive. So we have to fight our systems. And this is the platform that understanding the mysteries of the zoological world for creating a better future. And this webinar is the Lady Brabourne College webinar of Department of Zoology today. And my understanding is that a human disease, how we can connect that one. So that means students can understand that in a particular human disease, but that's a very crucial disease, critical disease, how we can handle that disease, what we have to know, how we have to handle it. This is a typical journey of the CSR with the critical disease cancer. That means CSR, not only CSR, the entire India, the entire world is thinking about that cancer research. Why the problem? Because millions of deaths. And there is no good medicine, but there are millions or billions of medicines are available. And there are the different cancers are actually thinking about the different disease. So CSR labs like IGIB, IICB, CCMB, IHBT, there are 36 laboratories. And that laboratories are working on different perspectives like the medical research, health research, not only health research, there are the engineering and the other chemical laboratories. So everybody try to find that how we have to handle the entire system. Today, in my talk, I'll concentrate on evolution of cancer. Then I'll go a brief idea of various cancers, cancer therapy, failure and opportunity, chemo resistance and recurrence that everybody thinking about that, how we can handle that things. Targeted drug delivery across the blood-brain barrier. So this is a specific issue, specific type of cancer, specific problem. So how we can handle that specific problem and delivery of nanomedicines. Before going to tell that story, let me tell you that about my growth. I was from a village. That is the first you can see. That is my village. Then I started my schooling at Jongrapara Bishekam High School. It's also in the village. Then I moved to a city like nearby city that Raja Piyari College, Uttarpara, for the BSc in biochemistry, in chemistry honors. Then University of Calcutta for MSc. Then moved to Cleveland Clinic Foundation, USA for cancer biology research. Then again moved to return back from USA and joined the Indian Institute of Chemical Biology under CSR for cancer research. But when I joined in 2007, that time there are very few cancer research taking place throughout the India. But nowadays, you can see that every year, many labs are joined to the cancer research. So at the beginning, that when we are a child, we just see the different pictures and try to understand what is that, what is that, what is that. Here you can see some of the pictures in the animal systems, in the plant systems. There are outgrowths. So there are outgrowths from the shape. So shape is change in few places, and that is called a tumor or outgrowth. And that tumor is sometimes benign in nature, sometimes malignant in nature. So benign is not the problem, malignant is the problem. But the thing is that we have to understand why benign, why malignant, why benign is not problematic, why malignant is problematic. Suppose you can give an example that if we can think about the prostate cancer, the prostate hyperplasia is problematic but not dangerous, but prostate cancer is dangerous. So if we think that how it is evolved, how the tumor is evolved in the body, in the plant body or the human body, it's a evolutionary dynamic disease, and reason why targeted therapy fails. And there is a selection process. So in our normal tissue, always there is a fighting mechanisms of the disease cells versus the normal cells. But the thing is, why it is disease cells? The disease cells means that cell it's changed from the natural behavior because of some thrust. What is that? Maybe the infection, maybe the maybe some kind of environmental factors, maybe carcinogens, maybe genetic mutations, maybe epigenetic mutations. But so for the disease, there is a change, and the process of fighting that the disease cells try to survive in that normal scenario, and normal cells try to kill the disease cells. But one day it is arrived. So the Darwin bypass is to prevent rather than cure. So evolution is helpless to eradicate anything. So the best possibility of all of the cancer researchers is to control cancers, turning cancer cell into a chronic sustained disease if unable to eradicate. And that is the possibility of eradication if at early stage. Here you can see in the right side picture that how it is evolved and how angiogenesis started to supply enough nutrient and oxygen to the cells which is growing in the normal scenario. And how it is happening? Let me tell one example out of several reasons. The example is the gene mutation that we always talk about that one. Doctors are telling that one, finding the genomic medicine. And we all know there are several genes because in a particular cell, 20,000 genes are actively expressed. But there are millions of genes are not expressed. They are present in the heterochromatin region. The silencing, desilencing is very important. One such tumor suppressor is called TP53, that is P53 protein. It is a tumor suppressor, but the mutant protein is oncogenic. The other tumor suppressors like RB, P130, P107 is a family of RB family proteins. That RB is retinoblastoma protein. That is a disease is retinoblastoma. And suppose P10, FOXO, there are many tumor suppressors, but these are crucial tumor suppressor. One such tumor suppressor, so one minus, so that means it is not expressed in the system. Then the evolution started. This is such an example of the hundreds of different reasons of the genetic mutations and cause cancers. In our systems, there is a biological clock in everybody's systems. As the clock ticks on, the intrinsic design limitations or trade-offs of our bodies eventually come into play. So we don't know when and where it will play. In who will get that kind of biological clock disturbance. So the thing is that we have to think that how we can handle the biological clock at the initial stage. So there is no general medicines that can handle that, keeping the natural systems always on. So in the early stage, everybody thinking about the genetic mutations and the phenotypic change that leads to a disease. When I was in USA, so that time I asked the questions that why not epigenetic, why genetic? But that time throughout the world, the big labs are actually jumping on that directions that epigenetic control is the major control. And that epigenetic changes leads to genetic change and subsequently affect the system. So I started working on in the direction, so where Budhaditya end up with that infection, the pathogen, and the host immune response, and there is a criteria of signal transduction pathways, and how the signaling systems are actually handling the immune systems. But in this lecture, I am not going to focus anything, but I am to try to visualize that how this particular disease we can understand and we can do something using the research and using the knowledge. So that time I worked on that particular scenario, that adenovirus, adenocarcinoma. So adenocarcinoma caused by the adenovirus, when infected in the cell, it is expressing viral genes and producing some proteins. The genes are like E1A, early. E means early. Early E1A, E1B, E2, E3, E4. These are 4 major proteins are actually handling the systems. And E1 is somehow in the nucleus dislodge the silencing complex from the promoters of the cell cycle genes. There are many cell cycle genes that is involved for cell proliferation. When dislodged from that promoter region, then what happened? The same E1A brings the other opposite player like the hats like P300 CPP, like PCAF, and bringing to the chromatin nucleosomal region, and it's changing the mark of the nucleosomal mark, the methylation to acetylations, and then the transcription started and protein expressed. That was published in Molecular Cell that time, 2003. And then I have to now, for the students, I would tell something about that our human physiological systems. There are multiple organs, many organs, from brain to toe. There are many things are there. But the thing is that you have to learn that one. Not a single disease. It's a multiple complex disease, and any organ can be affected. Like the bladder cancer, brain cancer, breast cancer, cervical cancer, childhood cancer, colon cancer, esophageal cancer, head and neck cancer, lung cancer, lymphoma, melanoma, multiple melanoma, ovarian cancer, pancreatic cancer, prostate cancer, sarcoma, stomach cancer. We have to understand at least something. So for the reasons, I am not concentrating throughout my career in a single cancer systems. I started my journey in different directions, and throughout my talk, you can understand what is my attitude and how we can handle the systems. Let us say leukemia. I never worked on leukemia, but I read a lot on the leukemia, because to understand that what is leukemia, how leukemia happens, then how we have to tackle the other cancer. Here you can see bone marrow that producing the hematopoietic stem cells, then multipotent stem cells, then myeloid progenitors, then ultimately differentiate to the neutrophil, eosinophil, basophil, monocytes, red blood cells, and other directions, lymphoid progenitor cells. But the problem is, because of that kind of some mutations, that some of the cells can get the empowered. They are empowered, and in the systems they are enhanced proliferations, enhanced survival power, so it's damaging the systems. Here you can see this is a single disease. Everybody think that one. Layman can think, oh, leukemia, blood cancer. But the thing is that even that one, there are 4 subtypes like ALL, CML, ALL, and AML, acute myeloid leukemia. So that means even each and every cancer, there are multiple subtypes. In case of leukemia, you can say 25% AML, 12% CML, 5% ALL, 44% CLL. 15% other leukemia. So that means that is not in these categories. So other type of disease. Say about lung cancer, smoker lungs versus non-smoker's lung. You can see the architecture and the color of the lung, but the problem is that some smoker, even at the age of 90, 80, they are smoking. They're the chain smoker, but they don't have cancer. But sometimes what happens, non-smoker, they are never smoke, but they have the cancer. What's the reason behind that? The problem is that 1 in 14 men or even 1 in 17 women, they are affected by the particular disease where drugs are not available, like pancreatic cancer. And there are stage 1, stage 2, stage 3A, stage B, stage 4. So when the disease is actually detected at stage 1 or stage 2 or in stage 4, it depends on that. The treatment strategy is different. Outcome is different. There are radiotherapy 0.8% because of the radiotherapy. The second-hand smoke 3%. Radon 0.5%. Other 6.7%. Smoking is 83%. But generally considered on the smokers are actually causative for this particular cancer. Whereas oral cancer, there are ulcers, there are patches, there are lumps inside the mouth, under the tongue or backside of the teeth, or anywhere, any place. But that's a big problem. And so for that reasons, when you have some pain inside your mouth, you have to visit dentist regularly. Examine your mouth, use SPF on your lips, maintain good diet, reduce smoking and drinking. Smoking and alcohol is the bad things. Everybody tells, but we have to maintain that way, how we have to use it. There is the female-based cancer, the cervical cancer. There are some cancers are the female-specific cancer, some are male-specific cancer. In the cervical cancer, this is another kind of virus. It's called the human papillomavirus. Like that, I worked 5 years in USA on adenovirus. Here, papillomavirus caused that things. This is the not only the virus, the multiple sexual partners, smoking, oral contraceptives, and the sexually transmitted this disease is actually problems in India, because 1/3 of the patients are from India. There are different tests, but sometimes the village people, they cannot go to the hospitals, or they don't know. So we need to avoid them. That the physical exam history, pelvic exam, Pap test every year, have to do this kind of test after the age of 50. Then HPV test, endocervical curtain, colposcopy, biopsy. So there are different techniques so that when can be identified that is it, or at what stage? Early stage 1B, late stage 1B, or stage 2B. There are 27% of new cases in the world, and there are 27% of the death due to cervical cancer in the world. It is in India. So 1/3% of regular death of cervical cancer is from India. Colon cancer. This is another problem. Mostly this is the food habit dependent problem, and the Western countries facing this kind of problem. So we started working on that colon cancer when I returned back to India and around 2009-10. There is also stage 1, stage 2, stage 3, stage 4, stage 5. There are the problems you can see. That in the colon cancer, that region is changing. That the architecture of the tissue is changed. And then the biopsy can tell us which grades it is. The stage 1 and 2 is around 40%. Stage 3 is 36%. Stage 4 or sometimes a recurrent stage is 20%. Recurrence means after surgery, chemotherapy, everything done, patient go home, after 1, 2 months or few years, again recur. What's the reason behind that? We know that stem cells can cause these things. I cannot cover everything, but I will try something that you can understand that. That is the colon cancer is the male, female, anybody can be affected because of the different food habits and the genetic makeup. Breast cancer, 1 in 8 women will be diagnosed with breast cancer in their lifetime. So it's a dangerous one. It's the second leading cause of death, second most common cancer. Incidence increases with age, so that all women are at risk. So please take your self-help. There are few different types, like a ductal breast cancer type, lobular breast cancer, mixed breast cancer, mucinous breast cancer, inflammatory breast cancer. We worked on the signaling mechanism and that tried to find out the marker. And 2016 onwards, we we just focused in the little bit deeper things, and the TNBC, triple negative breast cancer, it's dangerous than the normal breast cancer. So we worked on the triple negative breast cancer, and I tried to find out now that medicines are available, like tamoxifens, like other DNA damaging agents like cisplatin, carboplatin, those can be applied. But the problem is that in any cancers, this may not work. Ultimately, chemoresistance happens. So the opportunity may be something else. So I'll come to that particular area, that what's the real opportunity for each and every cancer. And there are some works published in cellular signaling, breast cancer research, cellular disease, and there are the therapy systems, and there are systematic therapy, the chemotherapy, hormone therapy, targeted therapy. Sometimes the hormone therapy didn't work. Next thing is prostate cancer. So it's not female case, it's the male case. It's a gland is very small in size. We are generally thinking that it's a very slow, sustained disease, and more men die with it than of it. So this is a dangerous thing, like the diabetes. Patients are dying not because of diabetes, because of the cause of the diabetes. That is the diabetic patients when actually affected by something else, another disease. And 1 in 6, so it is not less than the female breast cancer. This is more dangerous than the breast cancer. And there are, you can see in the screen, you can see that normal prostate architecture, whereas prostate cancer architecture is totally different. But the one thing is that there are almost more than 90% patients are prostate hyperplasia patients. It's not a neoplasia. Neoplasia case is very less, 5 to 10% maximum. But the hyperplasia patients also having problem because the kidney when filtrate the water, the urine cannot pass through the systems. So for that reason, there is a problem in the bladder. And there are the exams like the double diagnosis, the digital rectal exam, and PSA. That is full name is prostate specific antigen. So you can go for the test for PSA, but what happens, sometimes what happens, there's a range around 3 to 5. Some people, the less than 3, but it's cancerous. But some people, some patients, more than 5, but still not cancerous. So this may not be right test, but at least we can understand that that patient, the patient is prone to that particular disease or not. And there are better one came few years back. That is not PSA, that is PSCA, that is prostate stem cell antigen, not in India, but abroad, still it abroad. It will come soon. And there are staging like the T1, T2, T3, T4, different stage of the prostate cancers that can be ultimately get from the biopsy samples. Now move to another cancers. So I'm telling that some common cancer, some female specific cancer, some male specific cancer. Now it's another common, that's the brain, the terrific cancer. The brain cancer. It is the primary tumor types like that we are working on the glioblastoma, but there are several types: glioblastoma, astrocytoma, meningioma, pituitary tumor, nerve seed, lymphoma. There are other types. What is the problem? You can see at the left below corner that you can see 5-year survival rates. When you compare breast cancer, you can see 9 out of 10. When in brain cancer, thinking about 2 out of 10. When thinking about the glioblastoma, grade 4 glioma, 1 out of 10. Now you can think that the survival rate of the glioma patients compared to the breast cancer patients or prostate cancer patients or colon cancer patients or other cancers. It's a terrific one. There are the causes like the age, medical radiation, history of cancers, like the genetic history, and the mobile phone use. I am not sure, but the people are talking about that mobile phone can cause this because of the magnetic results. And there are several types of symptoms. You can read from the slides. There are people are confused. They cannot. There is a memory loss. There is several other problems. So we started this journey during USA when I stayed at USA. Oncogene 2005, and then here also 2013. Cellular signaling, FSG, molecular therapy, oncotarget, several others. These are some few examples that we are working on the glioma research since 2000, early 2000, and continue our research. So patients who have to do. The patients and the patient's relatives or family members should go for watchful waiting. It's the radiation therapy. Option is the hormone therapy, like the androgen ablation therapy for the prostate cancer, like the tamoxifen therapy for the breast cancer patients, some vaccines, surgery, chemotherapy, cryotherapy, but patient is always crying because of pain. The family is always crying because of mental pain. So always we are getting the pain from the patients if in a family there is a patient. So now let me talk about that. What is our platform? So based on that, this picture, our research platform I created here in 2007, the name of this laboratory is Signal Transduction in Cancer and Stem Cells. That is our platform. We are working on the signaling mechanism of a couple of cancers. We are thinking that whatever the signaling we understand, and we did some work, we are thinking about that because of the failure of the signaling pathway pathogens leads to chemo resistance. So maybe there are some crosstalks happen. So what are the crosstalks among the signaling pathways that we worked on? The EGFR signaling, Wnt signaling, NCOV signaling, estrogen receptor signaling, androgen signaling, and there are many, some other also. But the thing is that during working that one, we detected some of the physical targets, some of the crosstalk points, but still we don't have any answers because there are multiple issues. The thing is that in our systems, 20,000 genes are expressed. That 20,000 genes, after expressions, after transcription, translation, post-translation modifications, they are functional protein, structural protein, maintaining the architecture of the systems, either cell or that cell to tissue, like that kind of things. But functional protein, they are different functions, maintain different functions. Each and every function is important for the total natural beauty. And this is the protein level, the functional protein. Those are again changing their balance. Why that balance changing? And that may cause a disease. That balance is protein, when synthesized, matured, functions, they are destroyed by a system that is called ubiquitination dependent degradation. And there is opposite system is there, deubiquitination. Deubiquitinases are deubiquitinate to save the proteins from the degradation path. So that means these two systems maintain the balance. So we are working on the gene regulations and ultimately thinking about that, how proteins are post-translationally, they are maintain the threshold. If threshold are changed, then cause of disease. And that threshold is actually people talking about gain of function, loss of function, gain of function of the oncogenes, loss of function of the tumor suppressors. But the ultimate thing is that drugs, delivery of drugs, and the right drugs, right target to get the patient to the home. So we are working on the breast cancer, prostate cancer, glioma, colon cancer. Long time, last 2-3 decades. But our focus is deregulation of gene expressions in the disease cells and compared with the normal environment, normal cells. This is the epigenetic regulations at the level of epigenetics. So that means at the chromatin level, genetic changes like that, genetic mutations, that the different type of mutations, the SNPs, like the missense mutations, sense mutations, there are different types of mutations, deletion mutations, and transcriptional, post-transcriptional regulations, and translational, post-translational regulations. If we don't understand one such circuit, we cannot understand everything. That means ultimately what we are proposing. So the thing is that for the reasons, each and every student after completions of college and then university, then they are actually focused to go through abroad. Yes, they have to go. Either they can work in India, get experience, and always compare with the world. Never compare within the lab or within the particular institutions. Everybody have to compare that in throughout the world. What I learned, what I keeping myself is different than the others. How much I have to struggle. So I went to USA to learn the things that how yourself you have to improve from your mind and from your work. And then 1997-2005, after gaining a lot of experience on that cancer research in the different directions, different areas, I returned back to India to serve India in such a way that I have to apply my experience here. And I'm trying today again, I'm trying throughout my journey. So let me tell now, let me focus on that a particular cancers, the glioma, because that if I explain different cancers, maybe you all will be confused. Better concentrate on a particular thing in a glioma, not breast, not prostate, not colon. In some other lectures, I'll focus on those things and people can get the idea on that particular type of cancers. First of all, diagnosis and treatment. That is the first criteria for each and every patient. So in that directions, we started working when just returned back to India, and we observed that one, how we can diagnose a tumor inside the brain. So we used a technetium level probe we generated, in the high next slide, that peptide actually leveled with that radio level. So it's a radio medicine. We learned that one how intracranially we can implant the cancer cells inside the brain and produce a tumor. We learned ourselves, not using the stereotactic instrument, just by manually. Under the skull, we put that the rat systems we are using, the rat glioma cell line, that is C6 cell line, we implant it and watch for 20 days, 30 days. So we observed that one around 20 to 24 days that most of the tumor are dying. And when we are trying to see that opening the skull and getting the brain and seeing that one in the implanted region, what is happening? So we observed that one tumor is generated within 10 to 12 days. Then what happened then, we are keeping two batches, one control batch, one is the experimental batch. That means non-tumor bearing rats versus tumor bearing rats. We injected this medicine, radio medicine, through the tail vein, and we watched that one, taking that one whole body image. And you can see the top level is the tumor bearing rats image, and the lower one is the control image, 2 minutes, 5 minutes, 15 minutes, 30 minutes. You can see in the control, it goes to the body, given different places, and through the kidney, it's filtered out, and the bladder it goes out. But in case of the experimental animal, the tumor bearing, you can see that within the 5 minutes, 15 minutes, you can see there is a nice image in the brain because the tumor is there. So those cells are different from the natural, normal, differentiated cells because those cells are highly proliferating cells. So they are capturing more. And 30 minutes picture, you can see both are just similar. So that means radiomedicine is totally gone from the animal body. So it's very nice. Okay, this is a kind of diagnosis system. But the thing is that diagnostics is good, but we have to consider the treatment of cells. In each and every kind of cancer patients, the first option is surgery, then radiotherapy, then chemotherapy. But doctors will tell which will be the first, which will be the second, like that. In case of brain cancer, it is very tough for surgery because there are millions of nerves inside the brain here and there. So if one nerve is cut out, if suppose facial nerve, if it is cut out, then what will happen? The patient will be paralyzed. So there is a problem in the surgery like this normal tissue damage, high chances of recurrence. So why? I will come to that point after a few slides. Radiotherapy, the normal tissue damage, there is a development of radio resistance, and itself all you know that there is a cancer stem cells. Those are actually radio resistant. So by radiation you cannot kill. So you can kill the cancer cells but not cancer stem cells. And they can come back again. That is called recurrence. And there is a chemotherapy. That is also that off-target effect. So there has been toxicity in the entire body. So normal tissue damage, development of chemoresistance. There is a delivery problem, and you know why? I will tell you. Cellular retention problem, non-specific off-target effect. That is the problem of millions of drugs. That off-target effect, that means they are not targeted action. They have several other targets where they can work, but in low affinity. Let me tell you about which test can diagnose the brain cancer. The test for brain cancer, like the magnetic resonance imaging, MRI, like a tissue sampling for biopsy, like CT scan, improved one, the positron emission tomography, PET CT scan, a cerebral aneurysm, molecular testing of the tumor at the lab, clinical lab, or even research lab. And that molecular testings ultimately tell that what is there, what is the problem, how we have to address in the treatment strategy. In the left side, you can see the brain cancer, that is a brain structure. In the brain, there are many different lobes, and many different, the gray matter and the white matter, that there are different places, different functions. And the thing is that most of the common types of brain tumors in adults are meningiomas and gliomas. There are 120 different types of brain tumors. Now you understand, I just explained few slides back that 7, 8 different types. But actually, 120 different types of brain tumors. One such is glioma. And we are working on the glioma since early 2000. And there are more than 700,000 people only in the USA. Now you can think worldwide what happened. And I'll come to that point that the lifetime is just for the grade 4, it is just a year. There are different therapeutic techniques like the steroid therapy, chemotherapy, radiation therapy, and stereotactic radiosurgery. That is a very good technique. The stereotactic radiosurgery, that means that that you have to concentrate on all the rays to a particular point. So this radiation therapy is one such opportunity because surgery is the problem. But it cannot kill each and every cell, because I told you that cancer stem cells are radio resistant. So first we have to think about treatment factors. What are the factors? The type of tumor, size of the tumor, location of the tumor, growth rate of the tumor, overall general health of the patient. So these factors will tell that surgery is possible or not at all. Chemo possible or not at all. Radiation is possible not at all. So treatment options: surgery, chemotherapy, radiotherapy. So targeted drug therapy may be the ultimate opportunity, ultimate option. So our therapeutic opportunity may be a combination therapy, a combinatorial approach that multiple targets hit together. So synergistic effect will kill this particular disease cells. So targeted radiotherapy. How it happens? It discovered in 90s when I was in USA. In 90s, even it discovered in our institute, Cleveland Clinic Foundation, in the main campus, because Cleveland Clinic Foundation, they have 7 different foundation, different places, different satellite centers, like the Florida CCF, Florida Clinic Foundations. There are many such 7 such centers. The biggest one was the Cleveland Clinic Foundation's Cleveland, Ohio, USA, and that was 30,000 employees on that. And there, one particular center was gamma knife center. What is that? You can see. This is a tumor region inside the brain. And what happens? The instrument is designed in such a way, so all the gamma rays will come from different directions and pointed towards a single place, single point region. So this gamma rays, actually what happens? It's not selectively destroying. It says selectively that particular area is burned out. And this technology is actually giving 3 months extra lifetime to the brain cancer patient. So this technology is actually now applying for other patients like the breast cancer patient, other cancer patients. And like the chemo, there is a problem, and there is the thing that why we need the combination therapy, because the chemo resistance. So we may need some sensitizer. So that means, what is the problem? We have to remove the problem. So that means our challenge will be successful. So chemo resistance, to overcome that, we need the chemosensitizer. So combination. So chemotherapy generally involves the use of low molecular weight chemical agents to selectively destroy cancer cells, or at least limit their proliferation, while at the same time inflicting the least possible amount of damage to healthy cells of the body. So that is means off-target effect to minimize it. Suppose one, for example, the resistance to anti-tumor drugs is a major problem in cancer management. So we have to tackle that one. Mutant PBD3 bearing tumors often show resistance to a large number of chemotherapeutic agents used in the clinic. So we have to think about the PBD3 status, but we have to think about the other tumor suppressor status also. Several novel mutant PBD3 target genes have been identified that play a role in chemo resistance and oncogenic function of mutant PBD3. So side by side, we have to think the oncogenic mutant PBD3 versus wild type PBD3, what is the balance inside the system. Now, we have to begin again to understand the systems. We know that signal transductions, this is a process. And at the level of even in the higher secondary level, now students are actually learning these things. So signals can come to the surface, the reception of the signal by the receptor, you can see it dimerize, then it goes to that, transduced to the molecules, 1, 2, 3, 4 like such molecules, and then ultimately goes to the nucleus, the effector molecules, and it's attacking the nucleosome at the chromatin level, and as a result, what happens? That gene expression started. But if you see, in a particular cell, you can visualize the cell by using the microscope. In the naked eye, you cannot see anything. But inside that cell, the nucleus is again very smaller in part. And in that region, there are 20,000 genes are expressed, and they are talking one with another by different sending signals like that phosphorylation, dephosphorescence. There are many post-nasal regulations happen, so that means that some proteins are empowered, some proteins are again losing their power, like that kind of thing. So that means many things are happening. But the thing is that how we can remember that things? Just like the engineering circuits, so we can remember if we think one by one, if we read one by one and then connect, and then try to put our knowledge to understand that what is the beauty and what is the linkage, and how it is, what are the systems to understand that things. Like here, you can see little bit narrow. So only 4 different signaling pathways we can see easily. So when we can go to the one, then we can again beautify that one. What are the each and every molecules? Maybe in some pathways, 10, some 20, some 30 molecules are involved. So far, so as discovered, we know that things and we know the path and what is the effect on molecules. We understand that one, like the beta-catenin in Wnt signaling, androgen receptor in prostate signaling, like that NF-kappa-B, the guy is telling on that one, that inflammation and cancer, the infection that caused that one. Sorry, not cancer. His talk is a little bit different than the immune response on that host cell immune response. That P50, P65 that goes to the binds the NF-kappa-B sites. Like STAT3 sites are occupied by the RTK pathway. It's called a receptor tyrosine kinase. Then goes to inside the RAS, RAF, RAC, MEK, MEK, MAPK, the ARC1, 2, and then goes to the nucleus. And this is 3 different downstream again, like the RAS MAP kinase pathway, PI3 pathway, STAT3 pathway. Ultimately, what happens? So many pathways turn on so many genes, but naturally everything is under control and everything is natural. When there is specific problem in any of the pathways, the problem started inside the nucleus, and then ultimately outcome is. So cancer therapy and failure and opportunity is the ultimate things. Based on that, we'll put our research effort on that directions to ultimately save our people. CSR therapy. CSR has 36 laboratories like that CDRI, Central Drug Research Institute, IIM Jammu, ILCT, Hyderabad. These are the chemistry labs. There are many biological labs, 7, 8 biological labs out of 36. The chemists are actually producing many different type of chemical molecules like that, synthetic organic molecules. Again, from the plant source, they are producing the plant-based molecules. But the thing is that we have to test it, the anti-cancer activity. Then we have to test it, which cancer is affected. Then we have to test it, what is the target and how we have to deliver it. So in our lab, we are thinking about the not drug discovery, the drug searching based on the target, and then deliver it and get the outcome, better outcome. For targeted things, we are using the nanovehicles like that, exosomes, liposomes, other nanoparticles, PSG-based nanoparticles, liposome-based nanoparticles. The drug, the temozolomide, now goes to a specific drug, because there are hundreds or thousands of drugs are available. Even in the brain cancer, there are many, more than 20, 30 is in the market. But each and every doctor is prescribing this drug. It's called temozolomide, the commercial name Temozia. This is a little bit more costly drug. 100 milligram is around 12,000-13,000 and 250 milligram is 25,000. This drug, initially, it is affecting very nicely. Outcome is good for first initial 6 months. How it is achieved? That it can induce the anti-angiogenic power and promoting apoptosis. So it's working that, destroying that bad cells. But the problem is it failed. Why it failed? Because the chemo resistance appeared. So the thing is that doctors prescribe that one. It is actually failed from the phase 2 trial. Doctors prescribe that one, that this drug cannot be used to get, it is just alone. So we have to use 2 particular forces. One is radiation therapy and then chemotherapy together, and then subsequently only chemotherapy. So there are 36 doses have to take each and every brain cancer patient. The radiation therapy with the temozolomide. Every day, take 1 capsule of 100 milligram, like 13,000 every day, with the cost of radiation therapy. So every day, every alternate day, you have to take this combination radio chemo treatment. After cycle of 36, off the radiation therapy, but start the increased chemotherapy once a week, 250 milligram. That means 25,000 once a week. You can go. If patient can tolerate. Many patients cannot tolerate because of the toxic effect, vomiting, there are hair loss, there are many problems. Who can tolerate, that's good. But the problem is that after another 6 cycles, doctors will stop everything and watch for 6 months. But problem appears there that because of the chemo resistance, because of the recurrence, disease appear again. And for this particular drug, the chemo resistance appeared because of one such gene expression happens that is called a methylase, MGMT. This particular drug is a DNA damaging drugs. Like other drugs, other particular way of damaging DNA, like doxorubicin, like cisplatin, these drugs, but these drugs alkylate DNA. And this particular gene, which is actually expressed because of this pressure of the temozolomide, that gene is coming back, and what happens is removing the damage point. So that damage is recovered by this particular protein. So temozolomide is ineffective. So then the systems, the researchers actually going to that directions that producing the drug against the MGMT and making the combination therapy. But now next problem is that when I talk somewhere, doctors are there, doctors, even that my collaborator, Dr. Sandip Chatterjee, head of this Park Clinic, Park Nursing Home. His father was, Sudip Chatterjee is actually developed this Park Nursing Home, and his brother is endocrinologist, Sudip Chatterjee. This Sandip Chatterjee, my research collaborator, his wife also, Dr. Uttara Chatterjee, research collaborators from IPGMR in pathology department, IPGMR Kolkata, and that Mukherjee Chatterjee from his family, the wife of Dr. Sudip Chatterjee, also from IPGMR Kolkata, from biochemistry department. So we have a group, we are doing that kind of things, that kind of research in the clinical research. So our in vitro, in vivo, and then clinical combined, we try to understand the mechanisms. But here the problem is that this temozolomide, Sandip Chatterjee prescribes each and every patients of the brain cancer patients. But the problem, this drug cannot effectively cross the blood-brain barrier. Here you can see that when molecule goes inside the cell, what happens? Cell is telling, I'm sorry, but you are too highly charged, too large, not lipid soluble, you cannot enter the brain. This is a tight junction. That tight junction is created by endothelial cell and astrocytes tightly packaged on a basement membrane. So each and every drug cannot cross. So that means each and every pathogen cannot go to the brain. Each and every molecule cannot go to the brain. So some small molecule can go, but there is a problem is again. So that means that we need some that nanotherapy is the ultimate solution, and why I will explain a little bit later. Here, the temozolomide, what happens? We are thinking about that recently. We are thinking about that P68 is another player in our colorectal cancer research and also breast cancer research, and that is also happening in case of other cancers. So this is actually dragging that Wnt signaling pathway and other pathways. So we are trying to develop a peptide-based drug against the P68 interface from this TCF4 or other phospholipid binding sites. So that drug we can use for combination therapy with the temozolomide. So the ultimate goal of each and every cancer researcher is repurposing of existing drug molecules. So there are millions of drugs are available, but the thing is that we have to repurpose regular way to find out the best suitable drug for a particular target in a particular cancer. And then validation of the targets and the molecules by in silico in vitro. And then we have to think always about that minimize of target activity to reduce chemotoxicity and bioavailability. So here you can see there are many drugs are there. The platinum-based compounds, the DNA damaging agents like carboplatin, cisplatin, oxyplatin, the tamoxifen for breast cancer. For breast cancer, you can say the drugs are available. Lung cancer drugs are available. You can see the colon like iproribacil. Prostate, each and every cancer drugs are available. But the thing is that different drug, different activity, different places, different target. Either DNA damaging or protein target or receptor target, or even the antibody-based compounds are also used for this particular type of disease. Let me, with that, I am not going to my in-depth research area because many of you may not understand that things. that how we have to proceed that one. But here, such example, that one, we have to try first get the knowledge from that worldwide research scenario, what we know, how we can think about, we can create some hypothesis, and then design some experiments and then prove that one by using the in vitro experiments, then animal modeling, lung metastasis modeling, then clinical questions we have to address from the clinical sample study. So, and this is the thing is that currently we are doing that one, that molecular development, the peptide development, siRNA, small molecule, then combination therapy, to trying to think about that what is the target, what the molecules we have to select, what should be the combinations. But the thing is that one such combination may not be effective. So we have to sometimes change the combination for even the same cancer, because I tell you that even a cancer is a multiple types. For each and every patient, the type is different. All are glioma, but there are some difference. So based on the difference, we have to progress our things. So that means maybe we need personalized medicines for each and every cancer patients, but it's not possible. So we have to think about the possibility and the treatment strategy. So that means we can shape, at least increase the number of patients to go home. And currently we are concentrating on the last 5 years that next generation delivery of drug molecules as a nanomedicine to the destination inside the skull, that the glioma. So our goal is target identification for translation. We are thinking about the peptide therapy. We are working on the peptide therapy. We are working on the nanotherapy using liposome, PLGA, and the combination treatment using the targeted delivery of the molecules, so that molecules will be inside the nanoformulations, nanoparticles, either the hydrophobic, hydrophilic in nature. So we have to change the formulation type from the liposome formulation to the exosome packaging or to the PLGA formulations. But ultimate thing is that we have to add a receptor, that is the probe. That probe should be against the receptor that is specifically overexpressed in the tumor region. So in that way, we are thinking that we have to change the different drug formulations using the different combinations to understand the synergy and to save the patients. The problem in glioma therapy, this particular disease is highly infiltrative in nature. So within the brain, it is metastasized. Poor prognosis rate, and there is another problem. Any drug, when if it is entered, there is big glycoprotein overexpression, ABC transporter, so they are pump out the drugs from the brain. So that means if it is entered inside the cell, it is pumped out again. So the final concentration inside the cancer cells is less. Is there any solutions? Yes, there are solutions that I discussed. So TMZ, I discussed that one. So what we did, we designed a particular peptide against the SSTR2 receptor. That peptide we mutated. Actually, this is a small peptide. It's called the hormone, is the somatostatin hormone. 18 amino acids out of 18, 15 we mutated, one by one mutated, and then search for that one, which is the best mutant. And then we find out the mutant, that mutant peptide we ordered from the company. That peptide we coated on the nanoformulations. You can see right side, right corner, there is a nanoformulation. The PNGA nanoparticles, DIM is inside, and the surface is coated with this peptide. This peptide, again, we probe with the other things like that. To track this molecule, we have to track it. So we actually added some fluorophore like FITC, like Tambra, like biotin conjugation. So why? Because for different purpose, different tracking systems, we use this, again, the fluorophore attachment on the peptides before the surface attachment on the nanoformulation. Then we characterize that one by using different methods like TIM, transmission electron microscope, using SHIM, using atomic force microscope, zeta potential determination, size determination, shape determinations, everything. And we have to get the idea that molecules inside or not by using the release assay. So by these techniques, you can see some of the pictures like the PLGA, nano TMZ, nano temozolomide. This is the picture of the AFM picture, this efficient picture. Then you can see that the TLS graph plot, that AFN, how it looks like. Then currently we are thinking about the dual drug loading. So that means two drugs together. In the brain cancers, this is the strategy. that we have to put in the exosomes, because in molecular therapy 2015, we put it in the small peptide against the another tumor suppressor P10 and keep it inside the exosome by electroporation. So exosome carrying the peptide and exosome goes to the side-by cells. So that means it's a natural process, natural vesicles. We isolated from the cultured cells, then we electroporate our medicine, the peptide therapeutics, peptide medicine inside the exosome, then challenge to the tail vein in the tumor-bearing animals, and then develop that one. And this is the mechanism part. That what is the mechanism I don't want to discuss here because many cannot understand. So for the reasons, I'm sorry. And this is the exosome. What is that? In the normal systems, in each and every cell, they always release the microvesicles. One such microvesicles is exosome that can carry small protein, microRNA, RNA, and what is happening to the extracellular matrix inside the tissue. It can carry those vesicles, carry the internal materials and fused with the side-by cells or the distant places of the same tissue, and it's releasing that one. So that means it's a kind of cell-cell communication in the natural system. Like if you think about the plant system, the plant, they also share. The plant cells, they can form the plasmodesmata with side-by cell, and through the channel that one plant cell can transfer or release the material to the side-by cells, so helping that cells to grow nicely. Here in the electromicroscopic picture, you can see in the right side that these small red dots, those are actually the exosomes that is actually fused with the side-by cell. It is published in Molecular Therapy 2015. Here is the left side image, which is that the size measurement. We then analyze that one by trypsin digestions, and there are many experiments, in vitro experiments to prove that one, that our peptide is inside the exosomes, and it is colocalized with the exosome markers. Then animal modeling, we did that one, and we understand that yes, the molecule goes to the tumor region, and tumor gets regressed. So in that particular scenario, we design the peptide and then we simulate that one. And this is the, you can see intracranial tumor modeling. That tumor models, you can see on the rat tumor. This is the rat brain. And tumor, you can see there is a small tumor is there, but bigger than this cannot be possible because then animal will die. And then you can see the image of that region where the probes are actually going there. So we can deliver the targeted delivery of the molecule to the destination glioma. This is our combination therapy approach, the design of the experiments, how we have to do it. And here you can, there are two types of nanoformulation. One is exosome, another is the PLG nanoformulations. That too we are combining, and we're trying to challenge for the next level of study. Another type of systems that I talked about, the post-transfer modifications, that is very important. We are working on the directions that here you can see. We are working on the two different projects. One is that PTM means that acetylescence, phosphorylation, acetylescence, sumoylation, glycosylation, ubiquitination. So there are many types. We are working some of those. Methylation, like the TMZ can methylate that one. So we are working two important protein. One is CHIP, that is chaperone associated E3 ligase. So ubiquitination that destroy the proteins. And the other one is the Hauschp, that is actually saving the protein by deubiquitination. We are working, our one group is actually 3-4 students are actually working that particular area. And this is the lower part. This is our design experiments. That is, we are going to fund, we are trying to fund it. So that means DST CRB funded that program. Then this similar kind of program funded by the CSR. I will come to that point when I will acknowledge the funding agencies, that why they are funding these beautiful research areas. And recurrence, I will not discuss about these things, but everybody have to know because this is connected with that one. Because stem cell itself a lecture. My lecture is that totally on the cancer stem cells. I cannot go that direction. The only thing is that in the stem cells, their beauty is that they are radio resistance, they can self-renew, and they are, when cancerous disease conditions happening, that time it is very tough to destroy them, to eradicate them. So they can recur again after radiation therapy, after chemotherapy. So we have to more concentrate on that particular population of that inside the tumor region, in the hypoxic region, because these cells love the low oxygen tensions, the hypoxic core. These cells are staying. So it is very tough to destroy them. So we have to shut down. For the reasons you know that by the induced pluripotent cells research is actually going on last 2 decades, and that area actually Yamanaka actually they developed some particular systems, and as a result they got the Nobel Prize recently. And here is the Oxford Nano SOX2. These are the crucial marker of those stem cells. We are working on the nano programs in collaboration with Professor Siddhartha Rai, initially the ex-director of our institute, and then the director of IICB, and now is an emeritus scientist. But we are still together last almost 10 years. And we are working these cancer pathways, and the ultimate problem is the metastasis. We are doing the lung metastasis modeling, but the thing is that we are now going in the core things. So why metastasis happens? You can see the tumor cells are actually evade from the tumor region and going to the circulations and entering to the invade to another tissue. And for the reasons where tumor is actually from one primary sites to produce, to go, the travel throughout the body in another place, another tissue, another organ, and producing the secondary tumor. Generally, brain tumor, other tumors can reach to the brain, but the brain tumor cannot go out from the brain. It is residing within the brain, but in the brain metastasis is happening. So this is another important area. So we will focus on that directions. We're working, but we are not deeply working because we did so many things. But so we will not focus that one immediately. But we have a plan. And last 10 years, actually funded by not only India government, because this research is very tough. Huge cost is required in this kind of research. So fund, not only that, CSR has given me fund, DST given fund since 2007. So still they are funding me. Current ongoing fund is DST SCRB, DST Nanomission, DST SRB another fund. Now CSR is funding 1.13 crore. Just I got the letter yesterday. And a few years back, 2012, 2017, I had another program project, because I run the couple of program projects, international programs, that 10, 12 scientists from India and 10, 12 scientists from the USA or Japan. And now I'll tell that over Australia. So that Indo-US program with the Mayo Clinic USA, that's the, that best clinic in the US, one of the best clinic, like Clinical Foundation, best in the heart and in the heart cardiac disease, and number 1 in the world and number 2 in the urology. So we love to work on these groups. So it ended 2017, and there is a system biology program that our CSR, IICB, IGIB, and IICT from 3 labs, total 11 scientists together with the 12 scientists of the System Biology Institute for bioinformatics studies of that, and that systematic studies we did and completed in 2015 and then 2017. So these are the funding conditions, because we need crores of research fund to actually handle these so many programs, and to understand really in a particular type of cancer. If we don't understand the other cancers, we cannot differentiate that what is the difference between the other cancer to this cancer. And let me acknowledge, first of all, I always acknowledge my students, because they are the real actually workers. Day night they are working hard, they're trying to understand, they're sharing the knowledge with me. So they are sharing the knowledge, their knowledge to me, and they're getting the knowledge from me. They are trying to understand that one, how we have to handle the projects, what's the right design, what's the wrong design, what is the failure, what is the outcome. So there are almost 11 students almost completed PhD. Since 2013, they started to leave first student. So now 11 students got the PhD, and they are in abroad in different places. Even some are in India. One of them, Movita, is Imperial College of London. Shimana is MD Anderson Cancer Research. Firoz is Glasgow. Indonil initially went to Denmark, and then moved to USA. Kiran working in the Canada, Toronto. He is now applying for the scientist position. So that means they're mature, they're growing. So they will do something good. That is my vision. And current students, the Vinita, Nirajana submitted thesis, and they are actually, Vinita went to USA, Nirajana joined a very good editorial team in India. Gaurango, Shadodipa, Dipankar, Bhaskar, Saida, Rajni, Shabasti, Partho, Shani, Shubhajit, they are in the SRF and JRF. There are 3 more students coming. Dr. Shivani Sarkar, she is the woman scientist working in my group. And special thanks to Dr. Sandip Chatterjee, Dr. Suresh Bajoria, Dr. Uttara Chatterjee, Dr. Suresh Bajoria from NIH, Narayan Health, and R.N. Tagore. Dr. Sandip Chatterjee from the Partnernic neurosurgeon. Uttara Chatterjee, I told you that she is from IPGMR Kolkata, Department of Pathology. Siddhartha Roy, Dr. Porimal Sen, the chemists are actually my collaborators. Funding from CSA, DST, Indo, Japan, Indo, US. Now one more, Indo, Australia is now under processing that we depended. Now the decision is, we are awaiting for the decision. Last of all, not least, that let me thank, special thanks to all of you because of your patience, because you are listening my lecture. And the thing is that I don't know how much knowledge we can share, how much we can understand, but we'll try to understand because the internet is available. So anytime, any questions, if we throw to the internet, we get the answer. The thing is that right or wrong understanding, we need some expert. Thank you very much to all of you.
Thank you very much, Professor Ghosh. It's a wonderful session, and it was very detailed and covered a lot of topics. So it was amazing hearing from you. And we've also received an overwhelming response from our participants who are with us today. And I would again like to apologize to everybody that we are able to take only a very few selected questions, but we would provide his email ID in the chat box. So the rest of the questions you can either directly contact him, or we can send the questions to him as well. So, sir, so my first question to you is: can fibroadenoma lead to breast cancer? If yes, what are the chances?
What you are asking?
So, can fibroadenoma lead to breast cancer? And if yes, what are the chances?
I have no answers because there is a confusion regarding that things, the fibroadenoma. No response on that.
Okay, sure, sir. Okay, the next question we have is, what do you think about the use of alternative therapeutic agents like resveratrol for the treatment of human cancers? It's a plant derivative, as I believe.
Yeah,
so
it's a good drug. Okay. Any more questions? Then a couple of questions I can address later.
Sorry to interrupt. Chinu, could you switch on your video, please? Camera on.
Yeah.
Yes, sir, my camera is on.
Camera on, I think.
Able to see you. It's okay. Carry on.
Yes, sir.
I'm thinking that one, if you put a couple of questions together in next time, then I can address all the questions together in a single answer thing, right? But here, regarding these questions, the resveratrol. Resveratrol is a very good drug. It's a natural product-based drug. There are natural product-based drugs that are good for that. Because of that, there is no off-target, no chemotoxicity actually, no toxicity. But of course, there are the off-target effects are there. This drug, anti-cancer molecule, I can tell, that is working not a particular cancer. It is working in multiple cancers. And the problem is that it has the multiple targets in different cancers. So that means to understand the specific opportunity or outcome of that particular drug is very tough. So for the reasons, people are working with the resveratrol. We are also working with resveratrol, just recently started. The thing is that the selection is one of the important issue, that it may work from some patients, may not work some patients. Based on that, ultimately the thing is that patient perspective is the major issue for treatment strategy. So for the reason, some patients are getting benefited from particular drug therapeutic treatment strategy, some may not. So for the reasons, doctors sometimes what happen, changing the treatment strategy. So the strategy based on the outcome. So treatment started, watching. So for the reason, watchful, waiting, not for the patient and the family members. This is one critical issue of the doctors who are actually treatment started. that what is the outcome, everyday outcome, every week outcome, then change the therapeutic strategy is the one such important issue. If doctors are not focusing on that directions, then what will happen? The bad outcome.
Thank you, sir. Next, I have two questions together. Hello, sir, am I audible to you?
Yes, clear audible.
Often in stage 4 cancer, chemotherapy fails, increasing the rate of metastasis. Why is it so? And what is the effect of chemotherapy on cancer stem cells?
Yeah, it's a good question. The thing is that chemotherapy effect on, because you can think a tumor is a heterogeneous in nature. That is not a single cell type. In a tumor tissue, there are multiple different cells, populations are there. It is heterogeneous in nature, different type of cells. So the thing is that in that particular tumor, when stem cells are going to the progenitor cells, then progenitor cells, again cancer cells, so that means during such divisions, they also acquired mutations, right? So that means tumor generations, there is each and every time, there is a chance of acquiring different mutations. And we always know that in the particular, when the disease is mature disease in the late stage, then what happens? There are many cell contains many different type of mutations, right? So that means one such drug cannot destroy everything. So for that reasons, I am thinking that what was your original question? Please repeat one more time.
Yes, sir. Often in stage 4 cancer, chemotherapy fails, increasing the rate of metastasis. Why is it so? And what is the effect of chemotherapy on cancer stem cells?
Yeah, for the reasons I just explained that one. Each and every cancer is different. So that means common drug cannot be effective for each and every cases. But the thing is that most are working because all are anti-cancer molecules. But the affinity or the effectivity is different. So for the reasons, we cannot tell that one, all the 4 cancers we can treat by the same chemotherapy treatment or same chemotherapy regimen. We have to differentiate the tumor type with the regimen. One more thing is that we have to understand the patient system. Can the patient tolerate the chemotherapy or not? One more thing is that the chemotherapy, what we are trying to apply all 4, we have to think that what is the outcome in the next day, next week. Based on that, maybe we have to change that particular combination chemotherapy in different cancers. You got it?
Yes, sir. Sir, the next question kind of pertains to the current scenario a bit. The question is, does the anti-cancer mechanisms in bat get competed by some complementary immune mechanisms by the presence of so many viruses in them? So if you would share your thoughts on this.
Yeah, I understand. Maybe the Budhaditya will answer you a better way because he is working on the directions on the pathogens and the immune response, right? But the thing is that in the current scenario, if you think that one, the pathogen, we are fighting currently worldwide the coronavirus, right? And the coronavirus, the COVID, this is actually the novel coronavirus, not the original coronavirus that was actually happening in 2003. That is called the SARS-CoV. The SARS-CoV and this CoV, this not only true. There are MERS also. There are others also. There are the RNA viruses, and the thing is that these will come, go based on that treatment strategy. How we can handle these things. The thing is that what is the major concern in the system that if you go each and every hospitals, if you have the experience and if you think that one, the growth of each and every hospital last 30 years, you can see many hospitals, they don't have any kind of cancer treatment systems. But nowadays, they are actually developing the cancer treatment. There are cancer hospitals. Even some cities, there is no major cancer hospital. Like, for example, that Tata Memorial Cancer. I gave many talks in different cancer hospitals, even the Actec Mumbai. Now, the TMC Calcutta, I went there, gave the lectures. We have discussed with the Mamin Chandi, the director of the institute, as well as the Bhaskar Saha. He is the research director. So we have frequent meeting and we are exchange of knowledge of that kind of things. But the main thing you know that in the COVID systems, we don't know what will happen. Everybody thinking, every doctors, every layman patient, layman also thinking that comorbidity. That's the issue. Why the comorbidity is the issue? Because the thing, not only cancer, the other disease also, mostly in case of the cancer, if we think about, if we read the hallmarks of cancers in 2001 and 2011 that wrote by the Hanayan and Weinberg, Nobel laureates actually, they are actually developed that one, that the correlations of that, the cancer environment and how it is happening, the immune surveillance, immune suppressions. So as the immune suppressions happen in case of the each and every patients, mostly in case of cancer patients, if COVID attack, then what will happen? That it can change the multiple things. Initially, people think about that it is attacking the epithelial cells of the lung, but slowly they understand that one, that it is not only lung, it is damaging the other kidney, other organs, every directions, right? So the thing, we know how much the research progress, how much the outcome is established, and sometimes we have to re-research on that. Is it right or wrong? Like the vaccines developed parallelly, right? There are many countries are developing the three in trial 3, and people thinking that, oh, next month that the vaccine will come. Somebody thinking that, oh, there's a possibility of even the next year, because it is not so easy, because after development we have to test it, we have to go for the phase 1 trial, phase 2 trial, phase 3 trial, and these trials are systematic trials, would be from the FDA approval, but this is like a random trial. This random trial will not be benefited for long term, but short term it will be. So for that reasons, we actually, the people throughout the world, thinking about the random trial and get something at least to save us. Then we have to figure out that betterment of that particular vaccine. So maybe that couple of vaccines will come, and that vaccines will actually, something will be better than the others. Maybe in the down the line, maybe the next year, better vaccine will come, so we can prevent the disease. But the main theme is this comorbidity. It was there, it is now, it will be there. So comorbidity always, like that we always think about diabetic patients. They are always actually thinking about that. Oh, I should have the cancer, I should have cardiac problem, I should have kidney problem. It should not be. So for that reason, something is that we have to understand, we have to re-question ourselves to understand that one, which is right, which is wrong.
Thank you, sir. The last question I have for you today is, in case of the exosome-mediated delivery of peptides, how have you isolated the exosomes before engineering them? And what is the source of those exosomes?
It is a big question. It is a big question. To answer it, to address it, it's very tough. Not within a minute, but I have to try within a minute how I can address it. So exosome, it's a natural vesicles that each and every cell secrete. And this vesicle, like that one, that extracellular protein, that means after transcription, translations, protein are synthesized, one category of protein is called extracellular proteins, like the growth factors, hormones. So cells secrete those things. Like that, endoplasmic reticulum and Golgi, they are sometimes producing some vesicles, and that vesicles contains some material like that, mRNA, proteins, those things, internal material, and then release from there, right? The beauty is that these vesicles, exosomes is one category. There are multiple categories are there, and there are multiple sizes. Like that, some are very small, like the 30 to 50 nanometer, some are 50 to 100 nanometer, some are more than 100, some are more than 400. Even some are in the 1,000, more than 1,000 nanometer. The thing is that, what is the advantage of this? The nanotechnology actually provide us huge opportunity in not only in biological science, in the medical science, in the electronic systems, in the engineering systems. So technology is very nice. So we have to adopt the technology. Which technology is helpful for us? Like the gold nanoparticle, silver nanoparticle, there are other metal-based nanoparticles that is utilized for the tumor treatment, tumor therapy. So that means how they have to buy the thermal effect on that. So that means if we put the nanoformulations, you can accumulate in the tumor regions, then you have to use the laser or some radiation. So that means that particle will be produce the heat, and that because of that heat, cell will die. Here, PLGA and the other liposome-based formulations can be utilized, but these all are artificial, right? This is not a natural. But the artificials is FDA approved because there is a problem of toxicity of these nanoparticles. So that means exosomes, why we are choosing this? Because this is the natural systems, natural things. If we can put it back to the human body again, it will not make any problem. So for the reasons, we can take out the exosomes. So how we can take out the exosomes from the body? So let me tell you about the research things. So in the research things, we cannot do always the organoid research. Sometimes what will happen, in the cell culture model, the 2D growth of the cells inside our laboratory. And that 2D growth, what will happen, that when we cultured in our lab, that these secretory proteins and secretory vesicles, it will always secrete in the tissue, it will go to the extracellular fluid. In the culture lab, it is going to the media. That media, we have to do ultracentrifuge with that 1 lakh speed, 1 lakh G speed. Then we can get very small pellet. And then we have to test it, the size of the each and every particle. And then we have to test it, that is it exosome or something else. How we can test it? The research developed that one, there are some markers. Like PKH26 is the labeling system. There are some specific biomarker, sorry, not biomarker, the markers for the exosomes. That means exosomes synthesize that particular protein, and that is on the envelope, that marker is there. So that based on that marker, antibody testing by Western blotting, we can check that one. Yes, it is the exosome. One more thing is that this exosome can be re-engineered. That means from the tissue, we have to culture it, and that from the culture, we have to isolate these exosomes. This is the natural thing, right? But the thing is that during this culture, if we transfect with a recombinant vector that containing another factor, so another gene, that can be a peptide, small peptide. So that means when the exosome own protein is synthesized, that time the recombinant, the part will be expressed along with that. And that particular part may be a peptide therapeutics. So if we engineer in that way, then that protein when synthesized, it will, from one cell, it will go to other cells. But when we have to inject to the delven, it will go to the tumor. That time we have to make some targeted things. So it will carry and go to the tumor region and release there. And so that means that when the cell will take the materials from that exosome, that protein containing the peptide part, that peptide part is actually the molecule drug. So this is a kind of re-engineering, engineered exosome applied for the therapy purpose to damage the tumor cells.
Thank you very much, sir.
I don't know. Yeah.
No, thank you for patiently answering all the questions. Those were very different from each other. And thank you for being with us today. I would now request Professor Durva Bhattacharya to present the vote of thanks. Over to Professor Bhattacharya. And just 1 second before handing it over, we'll provide the email address of Mr. Ghosh in the chat box. So the remaining questions, and if any follow-up questions are there, you can directly ask, send it to Professor Ghosh. Thank you. Over to Professor Bhattacharya.
Tell you one thing. Before that, can I tell you one thing?
Yes, sir.
Because here it is that more than 200 participants, right? Or more than that.
Yes, sir.
So is it possible? Can you handle those questions and can you select the questions and send it to me? So that means you can get back to that and return back to them. Because
yes, we will try.
It's very tough to select.
Yes,
I don't have that much time. So better you can select all the questions, select the questions, send back to me. Then I'll return the answers to you and you will deliver them.
Yes, so we would try to do this if it's possible, because there has been an overwhelming response from the participants. We'll try our best. And thank you so much for your time. So I would now like to hand it over to Professor Bhattacharya. Thank you.
Good afternoon. I thank both our esteemed speakers, Professor Mrinal Kanti Ghosh and Dr. Budhaditya Mukherjee, for sharing their invaluable time with us and also being readily being a part of this webinar. I'm sure that their lectures has been very insightful and interesting to our audience. And by these lectures, they have offered just a glimpse of their wonderful work they are doing. I also take this opportunity to thank our principal, Professor Shivli Sharkar, and our IQSC convener, Dr. Indrani Chaudhary Dutt, for their support and guidance in conducting the seminar. I would also like to thank the head of the department, Dr. Sanjukta Mondal Parui, for conceptualizing this webinar. And also I would like to thank the other faculty members as well as our student volunteers for their help. I would also like to thank Arvind Prasad for their technical support. Finally, I would like to thank our audience, as without whose support, encouragement, this would not have been possible. Your response has been very overwhelming, and this webinar has provided us opportunity to remain connected in this very, very difficult times.